good morning to one and all. It is my immense honor to welcome you all to Day 2 of the National Conference on Genetics and Genetic Engineering. We hope you had a productive and insightful Day 1, and we look forward to another enriching session of discussions, presentation, and knowledge sharing. Before we start the session, I would like to remind you all about the quiz session that's going to take place today. Pay attention to all the talks and sessions to win exciting prizes. Now, let us kickstart the day with an enlightening plenary session on plant molecular biology, chaired by the distinguished Dr. Rex Arunraj. Now, to introduce the first speaker of our session, I would like to invite Rex sir on stage.
A very good morning, all of you. Today's speaker is a very important person. I hope you all had a very good meal yesterday, right? Lunch, what was it? Please, guys, what was it? Yesterday night, what was it? What was the source of that particular biryani? Can someone guess what was the variety that was used for that particular biryani? Louder, please. Basmati. Dr. Gopalakrishnan from Indian Agricultural Research Institute, who is the professor and head of the Division of Genetics, is the sole pioneering person in the introduction of 13 Basmati rice varieties. Anything that is named as Pusa Basmati is from their division, right? And I hope that no more introduction is essential. He has received lots and lots of awards, right? I have a list here on my mobile phone, but I thought that this 13 Basmati rice varieties is sufficient introduction for a person who is working or who has dedicated his life to rice genetics. And we both share the same university. In fact, a very good number of people here, we share the same university from where we come from, Tamil Nadu Agriculture University. And Dr. Parani was our senior. And Dr. Gopalakrishnan is a very good cricketer. He's a fast left-arm bowler. I have kept wickets for his bowling. I hope you remember that, Dr. Gopal. So no more introduction is necessary, and we are like lagging behind by 10 minutes maybe, right? So now on behalf of the department as well as my colleagues and student friends, I invite Dr. Gopalakrishnan to take the stage. You can clear all your doubts regarding agriculture biotechnology. He's a modern biotechnologist who converts or who integrates plant breeding with modern biotechnology, right? Thank you, Dr. Gopalakrishnan.
Thank you very much.
You can make it even in form.
Oh, well, that's good. Thank you, Dr. Rex, for this opportunity. I see Dr. Barney, Dr. Kiran, Dr. Devi, and Dr. Rex, and Iswari Reddy among the audience, who is a good friend from myself. First of all, I would like to thank the organizers for the opportunity. Some 20 years before, we were also students like you, attending conferences wherever, and good memories. Here, basically, what I'll be, I come from ICR Indian Agriculture Research Institute, which is one of the premier institutes dealing with agriculture research in the country. This is the library clock tower, and IRA has been the seat of Green Revolution. If you had heard about Green Revolution, wherein semi-dwarf varieties, the semi-dwarfing genes, basically RHT1 and 2 in wheat and SD1 in rice, they were sourced from CIMMYT and IRI to develop improved semi-dwarf varieties in the country. And this is our location in Bihar, where it was established in 1905. And this is the stamp commemorating the Green Revolution, which was, this stamp was released by the then Prime Minister Srimati Indira Gandhi in 1965. And we celebrated our centenary year in 2005, and this stamp was released during that time. And this is the division from where I come from, basically. After my undergraduation at TNAU in BSc Agriculture, I did my master's and PhD from the same division. And then for 5 years, I was in NBPGR's regional station in Jodhpur in Rajasthan, and then joined back in 2010. The bust in the front is the bust of Gregor Johann Mendel, which we inaugurated in July 2022 to commemorate July 2022, 20th July 2022. Does it ring any bell among the students? The answer is obvious. So the bust is that of Gregor Johann Mendel. So we celebrated the 200th birth year of Gregor Johann Mendel, and this bust was installed at that point of time. And this division, basically within the institute, this division has been responsible. It is we call it the heart of the institute, because any technology in agriculture, the main driver is crop, basically seed, and the seed comes from seeds of the improved varieties developed by this institute, by this division, has been one of the reasons for its great success in the past. This is the brief outline of the talk as such. I'll be briefly discussing about the rice scenario, and then we'll look into what makes a good discovery in terms of either the donor or mapping the genes or QTLs, and then how to translate into varietal technologies which can be taken forward, and the challenges. In India, roughly we have around 44 million hectares of rice, and the ones which are indicated in the dark green, they are highly productive areas, whereas as the color goes on lighter and lighter, the yield levels are on the lower side. In India, we have around 67 million farm families which are dependent on rice, and we use almost 30%, 1/3 of our irrigation water for cultivating rice. Last year, we produced around 170 million tons of paddy, which converts into 112 million tons of mill rice. Along with the paddy, we also produce equal almost. Can anyone tell what is the harvest index which we are looking at a crop like rice? Any idea? So it is roughly around 45 to 50%. So actually, we are almost producing 425 million tons of residues, and then and rice which is grown under puddled stagnant water, it is one of the major sources of emission of methane because of the anaerobic conditions. So, rice crop as such, it is estimated we around 3.5 million tons of methane is emitted from our rice fields, which was a talk of concern some 10 years back. But we are trying to address through research. How we are going to address is the question. If you look at it, rice as a crop is one of the most magical crops, I would say. You can see it right from Himalayas to below sea level cultivation, which happens in Kutanad and areas of the backwaters of Aleppo. So we have rice varieties or rice cultivars which are grown in almost all ecologies, right from more than 3,000 meters above the sea level to under the sea level. This is actually, there are uh, who is from Kerala here? Anyone from Kerala among the students? Okay. So what do you call these systems of rice wherein you cultivate below the sea level? Any idea? No. Where are you from? I saw a hand raised and it went back very fast. Where are you from? I'm from Kerala. Which place? Which place? Oh, sorry. Do you have a mic? Yeah. Malappuram. Malappuram. Okay. So I think Malappuram, I don't know, Malappuram is how far it is from Waitila, maybe, or even
Kutanad is in Alleppey,
Kutanad.
actually.
Kutanad. Yeah, it is in
It is
Aleppo.
like
Yeah,
7 hours to
7 hours, right. So Kutanad is a place, if you are venturing out, students, it is a very different type of rice cultivation which happens there. They call it the Pokali systems of rice cultivation, where water is literally, rice is grown below sea level. You cannot imagine how can cultivation happen below sea level. So that is what is happening in Kutanad region. And also, if you look into different ecologies, there are different kind of stresses, right from drought to submergence to salinity, all different kinds of stresses. So we have rice varieties, diversity of rice varieties which grows in literally any place across India. Briefly, we will discuss about the basmati rice varieties. This is among the rice varieties which is grown in India. Basmati is a special group of rice varieties which is known for its quality, and in terms of in the latest survey conducted by a food international group which deals with food, among all the rices across the world, Basmati has been rated the number one in terms of its grain and cooking quality and taste parameters. But the traditional basmati varieties, this is one of the traditional basmati variety which you see in the farmer's field. As you can see, this is somewhere nearing maturity. They're all very tall, like it grows 1.5, 1.6 meters. If fertilization is more, so because of that, when the panicle grain filling happens, it lodges. Invariably, it will lodge, and they are photoperiod sensitive, because of which rice is a short day crop, as you all know. So only when if it is sensitive to photoperiod, it will start floral primordial initiation only when the days are shorter, essentially after September in Indian conditions. And then because of this sensitivity, they were all long duration, like 160 days crop Even some of our traditional varieties, which is grown in Tamil Nadu, if you look at like Jiraga Samba, which is one of the popular short grain aromatic variety, they are all long duration. And they yield also somewhere around 2.5 tons, but they don't need much management as such, but the yield levels are very poor to make it commercially viable. It becomes very difficult if there is any other exigencies due to pest or disease incidents, then literally the yield levels can go even up to 500 kg or even 1 ton, which is not. So this is the picture of Professor M.S. Swaminathan, who has been awarded honored with Bharat Ratna Award very recently last year. He was the main architect of rice improvement. So what do we do? 1965, I said Green Revolution happened, right? Semi-dwarf varieties of wheat and rice, and we produced enough to feed the whole population. Maybe this generation would not know the how the PDS system, the public distribution system, the so-called ration shop, why it came into practice at all, because we didn't have enough food to feed the population. So we imported huge quantities of wheat before the Green Revolution happened from US, basically red wheats, which was not the most preferred for our cuisines or food which we prepare. So 1965 Green Revolution happened, and everyone was basking in glory. But Professor M.S. Swaminathan gave a talk on the celebrations which was arranged, where the stamp was released by the then Prime Minister. In his talk, he said that maybe time will come when other parts of the country also will produce enough food. So when that happens, Punjab need not produce rice for the whole country. Punjab need not produce wheat for the whole country. Punjab, Haryana, and the northwestern part, the so-called fertile plains, they need not produce for the other parts, because they will also be producing sufficient enough to feed themselves. So he said that possibly we will have to look into alternative mechanisms where the produce can get into commercial export quality rices. That is when he talked about the improvement of basmati itself in 1965. And the basmati rice, the rice improvement program in IRI owes to him. He initiated the program, and this was the first cross which was made in 1966 between Taichung Native 1 and Basmati 370. Basmati 370 is a traditional basmati rice cultivar. It is a germplasm collected. It is numbered as 370, collection number. So what do we do in normal improvement process is, the beginning is with the collection of all the ecotypes or the strains which belong to basmati category, and among them screen them, identify the best one and release it as a variety. So this basmati 370 was the 370th accession collected from across the basmati growing regions, both India as well as now Pakistan, erstwhile undivided India. And the crosses were made. But as I said before, like the international community of rice workers work on crops which are of international nature. But nobody was even willing to. International Rice Research Institute attempted to work on basmati rice, but they were never able to come up with improved strains or improved varieties. So with large number of students, master students and PhD students like you, the genetics of basmati traits were worked out. First of all, if you want to develop a product or develop a cultivar or a variety, you need to identify its unique features, right? So what is unique for Tesla? What is unique for Tesla? What is unique in Tesla? Am am I audible or there is no response I am seeing from the student? Anyone? What is unique for Tesla? Not now, but when it came, possibly. So he was able to think something which was really expensive at that time. He thought this would be the future in order to make a sustainable mobility system, right? So uh, Tesla came into existence because of that. But then if you look at this, like, you need to develop parameters for which you need to improve for, or what you are looking for. For example, in basmati rice, you have a minimum milled rice kernel length and minimum width. Then on cooking, how much it should elongate. It should have aroma. It should have minimum breathwise selling, because some of the rice varieties which you will see later on, they swell on cooking. So all these parameters were defined one by one, and their genetics was worked out. Uh, Professor E. A. Siddiq, who was there with Professor M. S. Swaminathan at that point of time, he hails from Ilayangudi in Ramanathapuram, and he, along with his students, looked at the inheritance of all these traits. And then finally, after 25 years, in 1989, Pusa Basmati 1 was released. This variety is the first semi-dwarf high-yielding basmati rice variety in 1989. And the duration of this variety is 145 days. It is semi-dwarf, and it is weakly photosensitive. And it yielded at that point somewhere around 4.5 tons. So 2.5 tons to 4.5 ton is a huge jump. Okay, unlike in um, other sectors, in sectors like agriculture produce, like paddy, you need a minimum volume to maintain the continuity. If you establish a rice mill which will have a turnover of 8 tons per run of around 4 hours, you have to have enough paddy to run it 24 hours and 365 days throughout the year. So that is the kind of volume we need, and this variety gave that volume for basmati rice. And with this came the establishment of modern rice mills in the northern part of the country, with investments from UK and also the machinery imported from Japan at that time. Following that, in 2003, we came up with another variety, Pusa Basmati 1121, and this variety is also 40 days duration, but the uniqueness of this variety is it has the longest mill rice kernel as well as cooked rice. So it has around 9 mm mill rice. On cooking, it elongates to at almost 20-21 millimeters. So this variety brought in another uh, paradigm change in which we look at basmati rice, and what you see in most of the wherever you are having biryani across the world, okay, you are having basmati, rest assured, okay, wherever across the globe, and this is the variety which made it possible. And then in 2013 we came up with semi-dwarf short duration basmati variety which matures in 120 days, and this saves irrigation water. It is photoinsensitive and having similar cooking kernel characteristics like 1509. So this is the, if you look at Basmati 370, this is how it looks, the paddy, milled rice and cooked rice. With PB1, we slightly improved the kernel length as well as the elongation. With 1121, both kernel length and elongation had a major quantum jump. And with 1509, we have been able to maintain that by reducing the duration, meaning that you can harvest the crop earlier, which means that the longer the crop stays in the field, the more riskier it is for the farmers. So we have been able to breed varieties which improved the yield from somewhere around 2.5 tons to around 6 tons, and from the duration got reduced from 160 to 120 in 1509. And I beg to pardon to Rex. It is not me alone. It is the whole team which did this. Professor E.A. Siddiq was associated with this development. He was awarded Padma Shri in 2007. Dr. V.P. Singh was instrumental in developing 1121 and PB6. He was awarded Padma Shri in the year 2017. And this is what we were exporting around 0.4 million tons in 1994 of basmati rice. Now we are exporting somewhere around 5 million tons. And the export earnings due to export of basmati rice is around 5 billion US dollars, which is around 54,000 crores annually we are earning through the export of this. This has happened not only because of varietal improvement, but also the state-of-art milling facilities and processing facilities. But the key driver for all these has been the varieties which were available, which could be processed and taken to the market. Now with this is a good achievement, which has happened only with conventional breeding. Make crosses, look at the plants, select good plants which are yielding, do the cooking, then select for possibly we do more cooking than any of our spouses do at our homes, because we on an average we cook somewhere around 1 lakh to 2 lakh samples per year to do the selection. So it was all done through conventional breeding process, wherein you make crosses, then look for segregants, and then identify and then do it. So, but then with intensive agriculture, when 1121 was introduced in 2003, because it was given higher prices per quintal, it is somewhere around 4,500 rupees per quintal of paddy. So everybody wants to grow. All farmers wants to grow the one which gives the maximum economic benefit. So monoculture had come into practice. Even for PB1 also at that time, it occupied the maximum area before 1121. So with that came the incidence of bacterial blight, blast, brown plant hopper, and sheath blight, which is normal when you do monoculture or some variety occupy in a large area. Then you have specific diseases which are associated, and all our varieties were susceptible because we didn't have any source for these diseases in basmati background. If you look at our exports to European Union, we were exporting the major share in the early 90s to only European Union. But then with 1121, it changed. We started exporting to the Middle East to the large extent. And then now our export stands somewhere around 0.2 million tons to European Union, mainly because the stringent measures which the European Union adopts. If you have heard trade barriers or cess, which is being or the custom duty which is to be paid. So basmati rice was exempted, was given duty derogation of almost $1,000 per ton. for imports in European Union. Even now it is there, the traditional ones. But then European Union came up from time to time, they come up with different parameters with which they try to reduce the imports. It is one of the import barriers which they exercise, and one of them is the tricyclosol residues, which they kept at 0.01 ppm. Which tricyclosol is one of the most widely used fungicide. It is very cheap, it is low cost, and it controls the neck blast incidence of neck blast to a great extent. So, but then they had so if you use the condition is such that if you use tricyclosol, it will get detected in the mill rice. So the imports also, because of this, when the consignments go abroad, if there is any detection of these pesticide residues, it gets rejected and the whole lot had to be brought back. So it involves huge costs, and so because of that, there is a declining trend in European Union. Luckily for rice, it is flooded with information from across the world. These are the genes and QTLs discovered for abiotic stresses, from drought to nitrogen use efficiency, salinity, heat stress, cold. You name a trait, you will get some gene or QTL mapped, which has been mapped in rice. Similarly, for biotic stresses, also the most important ones being blast and blight and sheath blight, and we have genes identified in almost all the chromosomes, and we have large number of genes or QTLs which have been mapped for other traits. Now it is a problem of plenty. Like if you don't have, that is also a problem. If you have also too much, that is also a problem. So what do you do? Although these have been reported, not all the sources of the resistant genes are available. If you look into literatures, like if it is mapped, if it is a gene which is mapped in China, you cannot access the germplasm or the donor for that trait. It is becoming increasingly difficult. So we had we prospect for donors within our germplasm. Our gene bank has around 120,000 accessions of rice. So we sourced these for disease like Bakane, which became very prominent after 1121 came into cultivation. We identified source. This is caused by Fusarium fugicori or Gibberella fugicori, which increases GA3 production in the plant. The plant grows taller and then it dries out because it uses all energy in growing up. We identified sources for all other traits which are listed. And then after identifying, you need to do mapping. So identification is the first step. Then you get into mapping, wherein you map the traits for example, here this is PB1121, which when it gets infected with Fusarium fugicori, it completely dries out at seedling stage. Whereas this genotype 1342 survives under inoculation also. So using this recombinant inbred line population from the cross between these two, we mapped the QTLs for Bakane resistance. Similarly, we have done for sheath blight. And this is one trait which was created in a collaborative mode with TNAU Coimbatore, wherein Dr. Robin, who was earlier the dean of PG school there in postgraduate studies in TNAU, he identified a EMS-induced mutant, which in a collaborative project where it was able to tolerate imidazopyr. Imidazopyr is a herbicide which binds to acetolactate synthase. So acetolactate synthase is involved in synthesis of leucine, isoleucine, and valine. So these amino acids are not synthesized because the herbicide binds to ALS and doesn't allow it to synthesize, because of which the normal rice plant dies. but the mutant has an altered herbicide binding site, therefore it can survive. So it survives, and we mapped the gene in chromosome 2, and then we have we also mapped the QTLs for reproductive stage heat stress tolerance. As you know, the climate is changing, and higher temperature leads to larger spikelet sterility. As you can see here, this is under normal condition PB1. This is chaffs, no grains. In the high temperature screening, you get lot of chaffs and very less grains in PB1. Whereas this donor, if you can see between both, in both the conditions, the number of filled grains are higher. So using that, we mapped the QTLs. And not only mapping QTLs, sometimes when you map genes, the genes are already cloned, and you identify novel alleles for the genes. This is a short grain aromatic rice, Sonasol, and this is 1121. So and we also have identified the alternate allele for semi-dwarf stature and also for lipoxygenase enzyme in case of rice. The lipoxygenase is important because during the milling process, the aileron layer is removed, which is which is a byproduct which is called bran. The bran oil is very quality wise, it is very high quality oil, but then you have to process it within 4 hours, otherwise it becomes rancid. So if you have a null for or the non-functional lipoxygenase enzyme in a genotype, then the rancidity will not set in faster. So that is the idea. Similarly, we have for low phytate also, and we developed the marker. And through molecular breeding process, through backcross breeding, we do foreground selection, recombinant selection, and background selection to introgress these traits or genes or QTLs or the novel alleles into the background. And we do a typical marker-assisted breeding strategy with phenotypic selection. As I said, we look for plant type and also the grain type. And this was released in 2008. This is an improved version of Pusa Basmati 1, which has, this is PB1 when it gets infected with Xanthomonas varaceae, Petovar varaceae. And this is the improved PB1. And we source the resistance genes XA13 and 21 from IRBB 55. And with this, the guidelines for testing the near isogenic lines also came into picture, and we created nils for blast pathogen also. Here you can see the kind of the donors which we get for blast. This is PB1, and you can see the cooked rice, and here you can see the brown rice color is red, and this cooking is very poor. So you'll have to introduce these genes from these donors without affecting the quality. That is where your markers come into picture, your background selection comes into picture. And we are able to release an improved version. This is because of neck blast, it is completely devastated, and we have improved varieties. I will rush because time is, I get an indication that time is already over. We have similar stories for QTLs for drought. PB1 we had improved for drought tolerance, and this is the kind of stress which we impose in order to identify the tolerant one from the. Here you can see the leaves are all rolled and it is starting to dry, whereas the improved version has better tolerance. And this variety under very high stress can PB1882 can give around 1.5 tons, whereas PB1 gives only 0.5. This is the variety which was released year before last. We have improved introgress QTLs for drought stress tolerance at reproductive stage into PUSA 44 also. So when you give drought stress, this is the kind of panicles you get, short and chaffy panicles, whereas it is well filled with these QTLs. Rice is normally transplanted, right? Rice is transplanted, right? So why is rice transplanted? Anyone have any idea? Why do we transplant rice at all? Anyone from the audience? Why do we need to transplant rice? Because other crops we directly sow. Maize is dibbled, it grows. Any crop, any other crop, it is not transplanted. Maize, palm millet, pulses, oil seeds. Why do we transplant rice? No idea. So this puddling and transplanting is necessary because rice grows in a wet ecosystem where the rain is very high, right? The moisture is also high. So under these conditions, the incidence of weed also is very high. So when you do this puddling operation and transplant, you see that the initial phase, the nursery is somewhere around 20-21 days. During that time, the nursery is already 21 days old. Rice plants are standing in the field, and there is standing water of around 3 to 4 inches up to 15 days or so. So till the rice plant tills and gets enough foliage coverage, there is no weed which can germinate under anaerobic condition, except for one notorious one, which is Echinoclova crusgully, which comes up, which is very similar, mimicking rice. So during transplanting also, if you are not well versed or expert, you can plant that weed along with the rice. So basically, standing water acts as a weedicide in case of rice. That is how this system of transplanting has evolved. And there is huge water resource, human labor involved, because it is all very process which has lot of drudgery. And if you don't do transplanting, this is how the field will look. Lot of weeds, and you will be very difficult to locate the. So with the herbicide tolerant donor, we introgress thus into PB1121, which when we spray with herbicide, it gets killed. This is unsprayed 1121. And this is the improved version with this mutant allele. Even after spraying, it can stand in the field. So that is what it can do. So far, we have released around 11 basmati varieties, which have been bred through introgression of genes for blight, blast, both together, then herbicide tolerance and drought stress tolerance. In non-basmati also, we have in the background of BPT 5204, blast and blight, and also recently in PUSA 44. Maintenance breeding is important. Once you release a variety, it is important to maintain the quality of seeds. So we do panicle to row paired rows progenies, and then look for the grain quality of the nucleus seed, and look for the presence of the gene, and then deploy it. Some of the challenges in translating the in-trait deployment is sometimes there are huge number of genes and QTLs reported, but we don't have access sometimes to all of them or any one of them. And the donor sources, because as I said, indicated during the red rice nature of blast donors, they have some linkage drag also sometimes, which is associated in case of drought stress tolerance. The plant height is associated with reproductive stage drought tolerant QTL QDTY 1.1. Cost is still a problem in case of background selection, and the critical mass of human resource, especially in areas like molecular breeding. At present, we are having the human resources, the students, the SRFs are getting trained in these areas, but then in other areas like genome editing, where we can create novel alleles for the identified functionally characterized genes, there is still a limitation. So there is mechanism for testing novel lines, genome edited lines. It is only available in rice, but not in other crops. And the compliance system, like we developed the herbicide tolerant variety, which was released in 2020. 2021 it was released, But then the imidazolpyr as a herbicide was not labeled for its use in rice. It is a herbicide which is most commonly used in soybean. So for use in any crop, it needs to be labeled for its use, and that is where we had to spend more time in terms of getting label claim expansion, and last year only it was available for cultivation to farmers. So these funnels, the breeding funnel, which we typically, the time taken for development of variety in the 90s, early 90s was around 12 to 13 years. With marker stud breeding, we have been able to reduce it to around 5 years, and that too introgressing some traits into popular varieties. But with technologies like gene editing and also with genomic selection and double haploids, we should be able to develop new varieties also in a shorter span of time. And the future varieties needs to be not only be high yielding, but also it has to have high resource efficiency. It has to be nutritionally rich, and also it has to be profitable for the farmers, as well as it should give the satisfaction which the consumer wants. These are some of the way forward, and this is one of the interesting papers which I picked recently from Development Cell, wherein these are popular cultivars, which are tree species, which has evolved through the process of endosymbiosis. They have acquired genes from the microbes as well as from the chloroplast, which helps them to overcome the variations in light intensity when it is growing. So this is one of its kind study wherein it can help in improving the biomass, and it has been proven in populace. And these are some of the concerns, including the sources available, the marker systems, and also the need for developing policy guidelines, especially in our country. We have clear-cut guidelines for genome edited products as of now, but we also have the guidelines for transgenics also, but then there is an impasse due to the cases which is running in the Supreme Court, which deters people from getting into this area of research. This is a teamwork. I am just presenting. As I said, there is a huge number of people, right from breeding to pathology to entomology, agronomy, physiology, large number of students who are involved, and the funding agencies like DVT, ICR. Finally, I would like to conclude with a tribute to Professor M.S. Swaminathan, who not only for us in IRI, he has been responsible for shaping the agriculture research at IRI, and for across the country also he has given so much in agriculture research, a legacy which we will be proud to follow. Thank you. Thank you.
Thank you, Dr. Gopalakrishnan. Now the session is open for questions.
Thank you for a wonderful talk, sir.
Thank you.
My question is, since you have identified a lot of residue genes in these paddy varieties, have you tried incorporating those residue genes in some other crops apart from paddy? Or if not, what do you think about the scope of the work, sir?
Yeah. So first of all, we have introgressed into our paddy varieties, and we also give the donor sources to paddy. But unlike for biotic stresses especially, they are very specific to crops. So if it is bacterial blight of paddy, it is happening only in paddy. The bacterial blight of cotton is something else. The strain is different, so it may not work there. So, but then there are crop neutral genes which can move from one to another. That is possible, but that is only possible through transgenic route as of now. But then there are genes like herbicide tolerance. The acetolactate synthase gene is across the organisms, across crops. So if you can mutate it using genome editing, then you can create genotypes which are tolerant to imidazolpyr or the imidazolone group of compounds. It is possible. So if you add where there are genomes available, it is very much possible. Otherwise, you have to take the transgenic route, which still has its own challenges. Yeah.
Thank you, sir.
It's a very nice talk.
Thank you, sir.
So, whenever you talk about basmati, people talk about aroma of the
Yeah.
basmati, right?
Yeah.
But now people claim that the basmati has lost this aroma. What would be the reason for those?
Yeah. There are several factors, sir, if you look into. One is the first and foremost is the time at which these crops mature. So the traditional ones, if you look at it, starts maturing in late October and it gets harvested in November. That is one factor. Whereas the improved ones gets harvested little early when the temperature is slightly on the warmer side. That is one reason. And farmers are also very clever. They grow the crop little early also nowadays because they are photoinsensitive. So you can grow them early and harvest it early so that you can take a wheat crop. Earlier the system was you grow paddy and leave the land fallow across the year. But now the crop intensification, that is another reason. The third important reason is nowadays the majority of the millers, the processing industry, they steam it or they parboil it before it is processed. So aroma is a volatile compound, and so the 2-acetyl-1-pyrroline, when it gets to steaming, most of the aroma is gone. And otherwise also, if you look at the demand globally, the majority of the demand is from Middle East, where it gets into all kinds of masalas. So aroma, you will only have to feel aroma when you take raw rice with dal or something. When it gets into biryani, it is some other aroma which you are looking for. So that is another factor which has contributed to it. But we are looking into it. There are other aromatic, short grain aromatics which have higher levels of aroma. Like we have Kala Namak, which is grown in the eastern part of UP, which has around 600 parts per billion of 2-acetyl-1-pyrroline as compared to 300 in basmati, in the traditional ones. So we are trying to look at the possibility of mobilizing those novel alleles from that so that we can improve the aroma also. But as I said, like everything has to come together, and also the way cultivation happens now, it is completely different from the earlier times. So you cannot, that is also one of the factor which has contributed to it. But yes, there is scope and we are working on it. Thank you, sir.
We have time for one more question. Yeah, the gentleman at the back.
Sir, what's your views on the direct seeded rice which the South Indians are growing nowadays? As I hail from Andhra, so I see my relatives growing like direct seeded rice more nowadays. Are there any benefits to it, or else it's the same as the transplanted rice which are growing in nurseries?
No. Direct seeded rice has several advantages. It has, but it is not that we are doing it for the first time. There are locations or ecosystems where they do traditionally direct seeding only, but we have to address the problems which are associated with direct seeding. Weed is one. And the second one is root knot nematode. Under direct seeding, it is more than the iron and zinc deficiency occurrence is more in direct seeded as compared to transplanted one. Uh, but if you look into the economics and the benefits in the long term, direct seeded rice is much better than the transplanted one, because simply for the reason that you saw the tractor which was puddling in the picture, right? That puddling operation alone takes up 30% of the water requirement. So if you can stop puddling and do direct seeding, you save 30% of water. And transplanting operation, you need almost 40% of the labor which is utilized in paddy cultivation is for transplanting operation. So you can save resources, and nowadays labor availability otherwise for agriculture is also low. So that also can be circumvented. Then if you look at methane emissions, the methane emissions are reduced by almost, because it is not anaerobic condition. Under direct seeding, you are not completely flooding all the time. So the methane emission also reduces by almost a factor of 50%. There is some nitrification, nitrate emission which increases, but it is not that alarming. And then you have the option for complete mechanization, which is not possible which is possible, but it has been tried, but it has failed. Like transplanting paddy using machines, it has completely failed. So it gives the option for complete mechanization of the whole process, wherein your dependence on labor for the purpose of planting, for the purpose of sowing, for the purpose of harvesting, everything gets circumvented through this process. So maybe 10-15 years down the line, you will never be able to see. Possibly those are the people who are the last crop of people who are transplanting rice. Nowadays, nobody, not even this generation, even people from our generation, you will not see them transplanting in the field. So direct seeding is going to become the way of cultivating rice in the near future. Of course, there are different ways to do it. You can dry direct seed, you can wet direct seed. There are different ways. That is a different topic itself. Yeah.
Thank you. Thank you very much, Dr. Gopalakrishnan. You have reached out with your rice journey. And anybody, if you have got any questions, you can talk to him anytime after the talk,
Yeah.
or even during the tea break.
Yeah, thank you.
We'll proceed.
Thank you, sir, for that illuminating talk. I would now request Dr. Kiran Kumar, Head of the Department of Genetic Engineering, to felicitate our speaker.
I request all the second year students to move to the Jina Hall quickly. Second years, please. And please avoid using the back doors and the front doors during the talk. Please, and don't talk.
Not there. We'll just directly over.
3 seconds. I won't take much time. No, I won't take. So let me start, okay? Good morning. I'm very happy to introduce Dr. Ishwaraya Ram Reddy. Dr. Ram Reddy is currently an associate professor at the ISER, Tirupati. He earned his PhD from Free University Berlin, Germany. After a brief stint of postdoctoral experience in 2011, he became a group leader and led a root biology team at the Department of Applied Genetics in the same university until 2016. From 2016 onwards, he started working at the ISER, Tirupati. With a strong foundation in plant root and rhizosphere engineering, Dr. Ramiredi's research focuses on developing climate resilient agriculture and exploring root-borne fungal pathogen host interaction and utilizing genome editing technologies to enhance crop traits. Thank you, sir, for accepting our invitation and delivering the lecture at our conference. Thank you.
Maybe it's better. No, mic on. Okay. I'm thankful for the organizers for inviting me over here. Am I audible there, or can I use a collar mic? That's better. How about this? This is better? Okay. So my lab works, I'm directly going through research, and then Dr. Gopal Krishnan is working on one aspect like getting to the farmers the seeds that they required, and then we were working under laboratory situations and then finding about the basic research. So we were looking at most of the time from the lab to the land, and then majorly my research is focused on particularly in the underground. We were looking into how the communication is happening below the ground system, and then what are all the players that are important for that below ground system so that the plant health or also the growth will be maintained. From the above ground system, we are asking a question that what is limiting them for increasing in the yield. For this, in this case, we always see that in a crop plants, particularly in fruit plants, we will get a lot of small inflorescences, then fruits, but ultimately we get very less number of fruits. What drives them, this mechanism? That is another biological question we are interested to address. And the other thing is where we stop right now, Dr. Gopalakrishna stopped at the DSR rice. So I begin that how we are developing rice varieties that are suitable for the direct seeded rice. So, so to begin with, I worked so long time on a phytohormone cytokinin that is very well known that it is a negative regulator of root development. If you can see over here, when you remove the cytokinin, then the root system growth is so fast, and then the architecture is also so different that it has more number of lateral roots and others. Using this basic finding and then identification of this one, we kind of engineer root system at our will so that we can increase the barley root system architecture and uptake of the mineral nutrients from the soil. We can increase the same thing to in a resource poor environment, let's say phosphate deficient conditions, and then again increase the phosphate deficiency either in the maize, or we can also look into the brassica napus or rapeseed oil. So in all the cases, we can modify the root system architecture in a way that how we would like to have it, and then useful for benefit of the crop, right? And then when we do this kind of a thing, later on when I came to India, then we thought of asking that question like, okay, now I know how to change the root system architecture, how to engineer them or tailor them to the needs of the particular soil. But if I get to know how exactly the root themselves know about what kind of soil they are growing, what kind of microorganisms that are there, and how do they communicate it is, then that is even much better. That the way that I design that root system is even much more better than looking for an overall view of from the increasing just by mass. Then I choose a particular root organ that is called as root cap. Why root cap? If you look at it, any kind of a root system, whether it is a dicotyledons or monocotyledons, when the root is growing towards the downstairs, it has a kind of a cap-like structure. That's why it's called cap. So it acts like as a drill. It will keep on growing like this, and then due to that wear and tear, and then it is protect the root meristem, and also this is the first kind of a tissue which perceives at the depth whether water is there or whether it is different kinds of elements are there, or some bacteria or other kind of fungi are there, or nematodes or anything. So we now know that the root cap itself loan like lot of tropic responses, the very well-known gravitropic responses, halotropic, nutritropism, hydrotropism, and communication with this one. And the root cap structures are so different along the land plant taxa. Then we ask that question whether we can able to understand this root cap growth and development per se. Then I just go through like how we will try to understand a bit of developmental biology, bit of physiology, then later a bit of pathology, and then finally genetics. So I will cover everything, a bit of it. The first part is developmental biology, where I would like to know how this particular root cap, this has a very short life cycle. If you can see over here, this is the root cap cells. Over a particular time period, automatically in a routine manner, it is always removed. The new means precisely these kind of cells in a particular time, they have to divide it from the quiescent center cell. They are differentiated it. After differentiation, they undergo kind of a programmed cell death. Then it will release this particular one. It's called as sloughing or border cell release. And now we would like to know how this entire mechanism is controlled and who is controlling this particular mechanism. So later I will tell you why it is important for the application point of view. Because I introduced you already, phytohormone cytokinin is important for the root system architecture negative rail. But when I see that phytokinin hormone is very much signaling, and then also the content is very high only in the root cap tissues. So we thought it might be do something related to the root cap growth and development. And then what if we remove the cytokinin very specifically only in that particular tissues where it is very high. So we develop a reporter line which is very specifically expressed in the root cap, and this is called as lateral root cap. Right? This whole thing is called as root cap. Now this is a two-dimensional, and you have to visualize it in a three-dimensional. It is surrounding this entire thing like a cap. Okay. So, and then it is present over here like this, and then we thought, okay, now in this particular domain, we will remove cytokinin content, or we would block cytokine signaling and see what will happen. So as expected, when we removed it, we thought of changes in the root cap system, but instead of that, we came to know that those plants are having higher root meristem activity, primary root meristem activity. So what does it mean? Means the root itself grows much faster because the meristematic region is increases. The cell division area is much increases. Now again, remember that what I did is I only remove the cytokinin in the root cap cells. But since the effect I see is in the primary root meristems. Then I ask the question whether within the root cap, whether I have any differentiation is happening. I told you that the root cap cells will division and differentiated and undergo to the programmed cell death. That division and differentiation is not happening over here, because if you see that differentiation, I can see them based on the Lugol staining, that means the starch granules over here, which is normally present only in differentiated cell but not in the dividing cells. But you can, then I also incorporated an EDU staining. EDU staining is, if you remember cell cycles in our thing, when it is incorporated during the cell cycle, and then a particular dye will incorporate it. When I see that particular dye in the cell cycle divisions, this dye will be activated, that indicating that these cells are under still dividing it. When you do not see that particular one, then that cell is not dividing anymore. It is only differentiated. It's only elongating it. Again, I show that the cell division and differentiation is different in these particular plants. And what other thing that I also noticed is when I remove the cytokine over here, the other hormone is activating so much, that is auxin. Not only that, now you can see over here in the two-dimensional and here, this division, like I show you in the video, it will release, the root cap cell is released like as a kind of a sheath. But in these plants, the sheath-like release is not happening, whereas each individual cells are coming like peeling it off. So before, a cap is like after some time, like 22 to 36 hours, comes like this. when the new cells are already there. So there is a rhythmicity. But now when I remove that one, that cap is not coming like that. Individual cells are coming at it, and then sometimes the release of the cap is kind of delayed, indicating that this particular hormone in this particular tissue has something to do with the how they are kind of undergo this kind of developmental program cell death, and then how they release into it. If you see further, the same thing when I do a 3D construction, like just to want to show you that how in 3D, like how this look like, in all these things you can see this sheath is coming like as a kind of a sheath, whereas here all over single cells are coming out of it. So why I was talking so much about it, now that is development, we were still finding out further one, what does it mean, how this is rhythmatically coming across, how a clock is controlling this kind of a thing. That is developmental biology. Now I slowly shift towards the plant pathology. So what does it mean? Normally when you are having this kind of it, when the root grows it, I prefer when you are not talking, okay? Being a teacher myself, every day I teach it, I like that people are not talking so much in the class. So when your root growing towards down, then this is called wear and tear, right? Because the root is going into the soil, friction is there, and then outer surfaces, outer cell layers, due to the friction, they have a kind of wear and tear will happening. Now, when you're looking into the soil, there is a lot of microbes are there. They're always waiting for an opportunity to enter into it. And then these epidermal cell layers on the root cap, they do have a kind of a structures which are all either some of cuticle layer, for example, the root cap, and then epidermal cells are very fortified with the cell walls. Normally, by default, plants are very healthy. That's why when you eat plants, we are also healthy, right? But then what will happen is, whenever these kind of vulnerable situations which are happening, these pathogens try to enter into that particular ones. For example, in the root cap when they're dividing it, or when the lateral root is emerging it break its own epidermis and coming out. So that then again there is a small pores are available. For bacteria it is sufficient enough so that they enter into the system. So they enter into the apoplastic region, then they go into the different kinds of it. Now we try to understand from development, from when we are having this kind of root club sloughing and others, what does mean. So when we look at it, we look for a particular soil-bound pathogen that is called as Ralstonia solanacearum, and then this is called as bacterial wilt. So the wilt pathogens, either it is a bacterial, fungal, what they do is they normally tend to go to the vascular system by crossing the different barriers. For example, they enter, and then from this enter from the epidermis, cortex, endodermis, pericycle, they will always go down into the vascular system. So when they enter into it, that is how they go. So we look at it. When these plants are treated with the Ralstonia solanacearum, we identified that particularly in the tissues where normally the weakest point of the plants, plant cytokinin biosynthesis, that means plant is making the cytokinin so high in the epidermal tissues and also in the root cap cells, indicating that plant is trying to protect this particular region whenever soil-borne pathogen is trying to attack. And then one way of their protecting them is they are synthesizing the phytohormone. Then we were looking into it, looking for the mutants that are lacking the cytokinin signaling and others. Then we count for how many of bacteria and others. We can say that when these plants are not able to sensitize or not able to do, sensitize cytokinin, then it is a kind of having a vulnerable effect to the particular pathogen, indicating that cytokinin is a kind of a positive immune responsive for in case of the Ralstonia solanacearum. The next thing that we look for it is, if that is the case, that means this plant hormone cytokinin has to regulate someone in these particular tissues. So that's a particular someone can be important for mediating a kind of a resistance. So how do I know that a particular tissue, I'm talking about a root cap cell layers, which is normally in micrometer in size. In cell wise, it is roughly around 100 to 150 cells, right? 100 to 150 cells in Arabidopsis. So what we did is we did a cell type specific proteomics, And then we take it for each biological replica at this many seedlings. So this one slide is corresponding to one PhD student of 6 years work, okay? And then they did a proteomics work, and then we identified small proteomics. Then we will also look for under salt stress, how this is happening. We also find a mechanism of how the root cap is mediating the salt stress. I'm not going into this detail, but what I was going to tell is now we will get to know what are all the proteins that are present only in the root cap. So among those proteins, we identified that some of them, which are belongs to the Jacqueline-associated lectins, very specifically expressed in the root cap cells. As you can see here, it's only in the root cap cells or in the lateral root cap cells, but nowhere else on the plants, and nowhere else on other roots or other root system. So we thought if these are all very specific protein in the root cap, are they under cytokinin control? Because cytokinin is synthesized in response to the disease. Then we see that these particular reporters are down-regulated when you are treating with the cytokinin. And not only they are down-regulated, and then they are mediated by the classical cytokine signaling pathway, indicating that this down-regulation is mediated by their receptors and transcriptional factors. Next thing is we look for it, this another protein, and we will look whether this particular protein, when you're treating them with the Ralstonia, a bacterium, at that time also, over a time period, that you can see a down regulation of it. So it is normally how they look like, they expressed in those root cap tissues, and then over a time period, hours post-infection, their expression is down regulated completely. Then what we did is, we generated a loss of function, knockout of that particular protein. We also generated overexpression lines, constitutive overexpression lines of this particular protein, and then again challenge this particular pathogen and see that whether this bacteria, like I told you, this bacteria is always wilt-loving area. That means it goes towards the, after crossing different cell layers, goes towards the vascular system. Now, if you look at it, this is the root cap, the bottom area. After infection in the Columbia, the bacteria here, I am tagged with M. cherry. So when you see the red color, this is M. cherry tagged Ralstonia. And then that bacteria enter into the wilt system, right? Whereas in the mutant plants, it is always, most of the time, it is always in the epidermis cells. It is still trying to enter it, whereas some are managed it. Whereas when I overexpress them, there you can see more number of bacteria already enter into the vascular system. When I go just above the elongation zone, another vulnerable zone, see the wild type, already the bacteria is in the vascular system, that is in the center. In the knockout, there is nothing, it is still in the epidermis trying to enter into it. Whereas in the overexpression lines, you can see a much more, indicating that these particular loss of function of these JAL proteins is causing a kind of a tolerance towards it. We will able to confine or confirm this particular behavior by measuring the log CFU, that means measuring the bacterial growth inside the root system, and also looking into the wilt index, so that whenever you see the mutant plants, the wilted plants are very less, whereas non-wilted plants are so high, so indicating that it is there. And then we again did the same kind of a thing on the soil, making a kind of a wilt index, drenches, keep on, but same fact from different assays, and then we confirm that knockout of this particular JALs is having a particular phenotype. Then we ask that particular question, then we come to the translation. We what makes them towards it, that basic research we are carrying out. Now, simultaneously, what we did is, these JAL proteins are causing this one. This Ralstonia solanacearum actually causing a very damage to the crop plants, particularly Solanaceae one, whereas in tomato, Arabidopsis is like a model plant, right? Then we thought to look for whether in this model plant, the orthologs of these JAL proteins are there, if there, and then whether they are responded to the bacterial Ralstonia, if yes, whether we can confirm it, their tissue specific expression, and then we started looking at them, and then whether we can able to do with the gene editing. So here in tomato also, 8 days post-infection, you can see Ralstonia enter into the vascular system. We are having 2 cultivated varieties, Arcavicas and then Hawaii as a resistant varieties. We show them the similar kind of proteins, the JAL proteins that what we studied in our Arabidopsis are also down-regulated here. And then when we are trying to make them Wicks means you are trying to create a knockdown approach rather than knockout approach. When you are using that particular one compared to control, those lines that are down regulated, they can show a little bit of tolerance, indicating again this is possible to translate Arabidopsis research in tomato, and then we are now looking into this particular one. What else we are looking at to understand underground communication? So when I say that if we want to know underground communication, it is important that at the particular point, at that particular cell, how they are making into it. So for this purpose, we develop a microfluidic system. So in this microfluidic system, what we can do is we can observe in a live microstream, and then we can also provide it either a particular bacteria for a case, in this case, for example, normally when a particular pathogen comes, depends on the pathogen, root hair numbers are increased or root hair length are increases, or the particular reporters are different. All these things we can quantify in live. So this particular micro fluidic setup can able to help us to understand not only the response of the root system, but also when we are using another biological control, such as, let's say we are using a particular pathogen, wilt pathogen, you are using Bacillus subtilis, you are using Trichoderma. So different kinds of things you are using it, and this is called as tripartite interactions. That means you are having a host plant, you are having a pathogen, you are having another beneficial one. Now we want to study that how their communication is happening, what makes them to talk each other, and then how come when three players are involved, how this is involving. We put it a metabolomics approach, and then we identified metabolites and others. So with this one, in the first part, I able to conclude you that it's important to understand how root system architecture goes. When you begin with the basic studies such as developmental biology and then able to look at it, that same findings has the ability or potential to translate into the field level using the crop plants and others, right? So in the rice system, I work on developing climate resilient rice varieties. We are having a mutational breeding, we are having a biotic stress tolerance, and then we are looking for the DSR rice. For this today, I will just more talk about DSR rice and just briefly mentioning about how we did a mutational breeding and identified the varieties that are all drought tolerant during the terminal flowering, right? Terminal drought. And then also for the disease susceptibility genes in case of the blast disease. And then we identified different hotspots of the Andhra Pradesh where we collect the samples over 3 or 4 seasons and identify some of the candidate genes repeatedly irrespective of the strains and others, or kind of differentially regulated to the pathogen. We now able to identify them. Then we did a similar kind of study in another blast, like somebody is asking whether the same pathogen is causing different diseases in different plants. We have wheat blast, we have ragi blast, we have rice blast and others. We ask the same question, whether the plants also have a conserved proteins to deal with this kind of a pathogen, if everything comes towards it. And then for that purpose, we did a transcriptomic studies between susceptible and resistant varieties of the ragi, and then we have meta-transcriptomic data analysis has been performed and identify the candidates among the rice, wheat and finger millet where they do have some kind of conservation in the way that they respond to the disease, and then identified some of the effector protein from the blast, and then the candidate R protein or NLR receptors from the rice, how they can be kind of responsible, which is conserved and then making into it. So these proteins we are now putting into the genome editing and then try to see that whether we can impart disease tolerance. When it comes to DSR, one student asking from Andhra Pradesh stating that many people are now adopting DSR compared from the transplanted puddled rice. So what are the advantages? So to save time, Dr. Gopalakrishnan already elaborated the advantage of the DSR and then why it is that is happening over here. So most northwestern part like Punjab and Haryana or time to time Delhi, you can see that there is always news headlines saying that water is scarcity and then they are stopping the water. This kind of water wars are happening within the Telugu, Tamil Nadu and Karnataka, within the Telugu states. We do have here about disputes about water sharing from the rivers, because rice is a very water loving plant, like that is how it's bred. And then over a period, and then all the varieties right now we are having is kind of acclimatized towards this transplanted puddle rice. In a nice infographic, Dr. Gopal Krishna show you that in a different ecosystem how rice grows it. But from all the ecosystem, we kind of, most of our varieties are kind of improved and bettered that are suitable for the transplanted puddle rice. Now the water scarcities are so high, and then we want to go for a direct seeded rice. Government is providing incentives in order to encourage the farmers to go to the direct seeded rice. Everything is good, but then in order to put in the direct seeded rice, so far they are having in a very luxurious conditions. There is no weeds, there is no competition. Then there is no need of increase the root system architecture to go deep, because everything you are providing over there. So we were asking that question, seedling vigor index, that means establishment of rice seedling under direct seeded conditions compared to the transplanted one, and then having an improved root system architecture is one of the key parameters for and then to withstand or to outsmart the other kind of things. Of course, there are several other problems associated with the DSR, but the benefits associated with it, when we can able to address those negativities, then it will helpful or soon a reality that we can go for a direct seeded rice. So we are trying to address that negativities. One of them is improved root system architecture. What we did is, we take 200 rice varieties, and then these 200 rice varieties are very completely belongs to the program that are developed by from TNAU, AP Angru or IRI within India. It's not from TK panel or something. And these 200 rice varieties normally grown in our areas, and then we test them in 3 different conditions: laboratory conditions, wet DSR, that means water is not completely removed, unlike not, and then where is the dry DSR, only the water is provided whenever it is needed, like line sowing. Then we did a genome-wide association studies. So we look for 15 different traits, starting from the root number, root length, root weight, shoot length, shoot everything, 15 different traits. Then we identified candidate genes. Then those candidate genes, we are trying to validate it, their role directly involved into the suitability for the direct seed rice using gene editing. And then we are also, on one hand, we are also using them identification of the markers, and then using marker assisted backbreeding. So this is how we are doing. This is a schematic, and then I'm just quickly go through it just because whatever I saw is the results only. We did this many different traits, correlation graphs. So wet DSR and dry DSR, seedling vigor index 1, normal distribution, how this is look like. And we identified so many common QTLs across these 3 different conditions. And then we identified them of those common QTLs in 3 different conditions: 7th day, 14th day, and 21st days. And genome-wide association studies, I can show you here, whether it is in the wet DSR and dry DSR for one particular root right, exactly in the same chromosome, similar positions, we see a kind of very strong associations indicating that that particular genomic region or the gene might be directly involved into that particular trait, whether it is a root number or root biomass. So we prioritize the candidate genes from the QTLs, and then identifying them, then we are looking into it. While we are looking into it, the same gene population has been evaluated for the lodging resistance, that means culm strength, and also for anaerobic germination. And we identified 2 parents, that is MTU3626 and BPT4004. And then which are all having on either side of the spectrum. One is not suitable for the direct seeded rice, but is very good marketable range, because it is BPT5004. Samba Masuri, it is a very popular variety in the southern part of India. And then this MTU36. So we kind of reaffirmed this one, and then we make it into a backcross method. And then these particular crossings will allow us to identify the lines we have based on our already published one, and then our own one, we make the markers, polymorphic marker, which are different between these 2 parents. And then we developed the new varieties that are having both early seedling vigor traits and grain size, which is a trait for the BPT that we are doing it. So within a time I am completed this work, which I gave it to you very fast and in very nutshell, try to convince to you that in general, when you ask a particular right question and then look for it for this one, then there are ways now to translate into a kind of a product or particular one, and then the time taking to reach to the product level due to the contributions from the IRA or other kind of agriculture research stations and all the people that now is going reduce and reduce. So there is a hope, and then that hopes lies in you people who are younger generation to take forward this one into the much more suitable condition. Thank you. This
thank
is
you.
our new campus, so please do visit us. And then we are having a vibrant programs of BSMS, professional masters, PhD programs, and of interdisciplinary possible.
Thank you, Dr. Ishwar, for insightful talk. So we can take a couple of questions.
Please. Because time is running out, and then in order to catch it, I also went so far in speaking. So,
Good morning, sir. Myself, Prasanna, I'm a PhD scholar. So I'm also working on a root system in legumes. You mentioned about the chain architectural system related to the genetic expressions and all. So there are reports like the root system can be changed by the micro and macronutrient supply to the plants.
yes.
So is there any relation between can nutrient supply can change the genetic expression level in a plants? Would that be the reason for? Would there be any correlation between of those two
So
aspects?
it's like the same thing. Like when you take food, when you take less food, we will be ill. When we take too much food, also we are ill, right? And then similar thing, plants do have, root system do have a kind of a mechanism to detect whether they have enough nutrients or not enough nutrients. So they only take optimum kind of a conditions, like whatever they require, they take it for their growth, even if you supply so much. And sometimes what happens, some nutrients when you supply so much, it can also cause toxicity, right? And then the plant root system find a way that to identify very specifically whether this is very low or high, and then there is a sophisticated mechanism to find them phosphate is deficient or nitrogen is deficient, and then they can able to see that even a change in micromolar concentration or nanomolar concentration, they can tell it whether the nutrient is available in the millimolar or micromolar or grams, like molar concentration, how much it is there. It is still able to tell it, and then it changed architecture such a way that to adjust its behavior towards this. So that is a possibility, and then lot of literature, lot of research has been done already in that direction.
Thank you. Thank you, sir.
Thank you.
I have one question. So when you knock out a gene responsible for cytokinin, any other hormones would be affected?
There is always an interplay between the hormones, because when I remove one of the major hormone, and then obviously the other hormone come into the rescue, or they always alter a lot. So that's why we remove it very specifically in a specific time period, and then also very specific tissues. And so that determine whether I'm removing the cytokinin only in the root cap, or when I'm removing the cytokinin only in the shoot apical meristem or inflorescence meristem. That changes a lot. So if I remove cytokinin completely all over the plant, it can lead to the negative effect also. So when I remove the cytokinin or add the cytokinin completely to the plant, again it has a negative effects. So understanding about its role in that particular stage and development in that particular tissue is the key. Once you understand that particular one, then you can modulate it. So the root system architecture I showed in earlier in barley, maize, or all these things, or only cytokinin is removed very specifically in the particular parts of the root system, not even in the shoot. When you are able to do that, then it is able to make into it, right? Thank you.
I have a final question.
Please.
Because you are modulating the root cap as such,
Yes.
root cap gives you a physical strength to poke through the soil or any other hard surface. Did you see any variation in the the manner in which the rice establishes the root system because it lacks the hardened root cap.
Yeah. So far the research related to the root cap is very less, and then that too happening only in Arabidopsis plants. And then related to the crop plants, particularly to study the soil compaction, how much it is able to do it when we are remove the root cap, whether it is still able to penetrate into it or whether it lost it or not, that research is not done. We are now trying to do the same thing in rice and then try to see that how now when I put it into the transplanted condition or in a normal wet DSR or dry DSR conditions, whether how good or how important is this root cap to have intact and then to penetrate into it. Otherwise, what will happen towards it? This kind of thing we are now looking into it. It's a good question.
Thank you, sir. It's a great work translating from Arabidopsis to rice. Thank you.
Thank you, sir, for that enlightening talk. I would now like to request Dr. Kiran Kumar, sir, to felicitate our speaker.
Students, hi students, there's a small change in the schedule. So we'll break right now for refreshments outside for 10 minutes. We have preponed the poster session. So from the poster and the oral sessions will start in parallel. So session 1 is now going to be from 11 a.m. to 12 p.m., which is in the next 10 minutes. So please put up your posters in the atrium. And session 2 poster, which was from 3 to 4 p.m., will now happen from 12 p.m. to 1 p.m. Okay? So the entire poster session is from 11 a.m. to 1 p.m. Oral presentation students, please assemble in this hall and GNR because it will happen at the same time. Okay? If you have any questions, you can ask me or any of the volunteers directly. Thank you. We can now break for refreshments.
Good morning, one and all, especially the judges as well as my companions who are here to listen to the talk. The topic I'm going to present here is genome-wide identification, functional characterization of protein phosphatase 2A gene family and its role under abiotic stress in Cicer-aratinum. The cultivar that I'm using here is ICC 4958 and under the guideship of Dr. Neelam Mishra, and I pursue my researchship in St. Joseph's University, Bangalore. Okay. Yeah, first we come to the introduction. Chickpea, that is Cicer-aratinum, is said to be a cool season crop, mainly grown in winter, especially in the Southeast Asia as well as the Mediterranean region. Mainly limited by abiotic stresses such as drought stress as well as the salt stress. These stresses are mainly seen at the terminal stage where the plant starts producing fruits and the flowers. And the most important thing is it plays a major role, or it constrains its yield in the chickpea, accounting to about respectively losing about 50% and 20% of the yield losses globally. It's not only in India but also globally when considered, according to the report of the FoodStat, that is FoodStat. Now here basically I've put a picture what happens from this abiotic stress. The plant usually loses irrespective of drought or heat stress or salt stress. The plant starts wilting. Now basically the plant has stress responses, that is the ROS signaling, that is reactive oxygen species within them. Certain time what happens when the stress is too much, the plant too decreases its activity, that is a photosynthetic activity or any other activity, and then this will further lead to the decline in the yield as well as the productivity or the yield that is required for the market. Okay. Now, before I go into what my study basically holds forth, I have conducted a review of literature. Now, in this review of literature, under the plant site heading, we see there are plenty of plants that have been used, mainly Arabidopsis, Glycemax, as well as you see rubber, and basically, generally, they have considered a review paper of all plants, but the role of PP2A genes in chickpea is not being highlighted anywhere. And that's the reason this has pertained for the interest of my study. And that's the reason I would like to carry out forward. Okay. Now here I come to the hypothesis. The PP2A gene chickpea plays a significant role in improvement of drought and salt stress tolerances. And to prove these, I have my objectives here. Firstly, the identification and functional characterization of PP2A genes in Cicer-aratinum using genome-wide identification studies. Secondly, after I finish those, I have done the expression analysis of PP2A genes in chickpea under drought and salt stress in both the tolerant and susceptible cultivars by NGS, that is next generation analysis sequencing and quantitative PCR. Okay. Sorry. The work plan and the methodology that I have used, here are certain tools that I have given. I have not shown all my results, but these are the bioinformatic tools that I have used. First is Mega software, then GSDS, that is Gene Structure Display software, the Meme Suite software, TR Rosetta, and Heat Mapper. Then secondly, the expression analysis of these plants under control and stress conditions have been carried out through real-time PCR. I have used the NGS, that is NGS data analysis as well as the real-time PCR. Now here I would like to show you my results. Basically, I've constructed a phylogenetic tree where I have taken 5 species, including the chickpea. It would be 6 species. Now these 5 species, when constructed under the phylogenetic tree, that is using the Mega software, I have used the neighbor joining algorithm and consider 1,000 bootstrap replicates. Now this here basically I want to say that the result I want to say that is PP2A genes have been having 3 subunits, that is A, B, and C. Now here basically these have been separated based on their families and B basically on their subfamilies. So we see PP2A A subfamily, PP2A B dash subfamily. So there are 6 clades that have been divided into, and here each of these families have been if we see the phylogenetic tree very carefully, these have been closely related with the Arabidopsis species, which is considered as a model organism that have been used. It could be even certain chances, as it's a pulsus, it might be even closely related to the Lotus japonicus legume crop. Next, as I finished with the phylogenetic tree, I go into the motif analysis. Now here what happens is, basically we see that 2 to 8 motifs have been expressed in most of these species. In chickpea as well as in Arabidopsis when we compared it to. But basically, if I identify one thing, that is 80 PP2A tone family, that is in both, only one of the family or one of the motive has been displayed here. So that is basically or generally the highlighted difference that we find. In GSDS, that is gene structure display here, basically we see that the exons here are it's in a very huge constraint when we see according to the regions when compared to Arabidopsis. Whereas the unrelated proteins might be of the similar size, but the exons seem to be a little larger when it's divided. Here we have seen that there are 21 genes that are being obtained of PP2A, which have been evenly distributed on 7 chromosomes, though it's diploid, but there are only 7 chromosomes where these 21 PP2A genes have been expressed. Finally, we have used the TR Rosetta to identify the protein structures, the structure of each of these 21 genes, and then we have used the PyMol software to superimpose these, to see the similarities between the structures. And basically, out of these 21 genes, we found that 4 of them were superimposing with each other, mainly PP2A A1 and CA, that is chickpea A1, as well as the PP2A A2 and A2 of Arabidopsis, as well as the A3 with A1, as well as the A3 with Arabidopsis A3 with A2. Basically, expression data, I want to say that there are 5 genes that have been expressed both in drought stress as well as in salt stress. And finally, the expression data that the real-time PCR that I've done is basically for the susceptible variety. The relative expression of the selected PP2A genes have been carried out, both with the susceptible as well as the tolerant one. But because of the time constraint, I have only expressed one of the data here, which shows that PP2A genes really play an important role in in controlling the drought stress as well as the salt stress. Finally, in the summary, I just want to say there are 21 genes identified, as I told, and these are equally distributed by using the map gene to chromosomes, and 9 out of 21 genes were upregulated in salt and drought stress by quantitative qPCR. The scope of the study is, the study aims to comprehensively identify and characterize PP2A genes in chickpea and to explore their functional role in abiotic stress tolerance, which will improve the resilience of the crops in drought and salt stresses. So these are my few references, and I would like to thank everybody for giving your kind attention to me. Thank you, one and all.
So sorry, any one question from participants? No. Sir.
So they are phosphatase genes. So what is the function of these genes in abiotic stress?
So this will withhold, it will enable the ROS, that is the reactive oxygen species signaling. It will enhance the plant. The plant will be able to enhance it, and then it will be able to improve its yield. When giving the yield, basically when I showed you in the review of literature, one of the plants of Arabidopsis says that PP2A-B double dash has shown that ROS signaling, it has improved the ROS signaling, and which has finally given, an emphasis of increase in the drought
What is the
yield.
link between phosphatase and ROS generation?
Sir, it helps it it plays a direct role, might be in the pathways. I have not concentrated on the pathways, sir.
What is the source of these genes you have taken from the?
Yes, ma'am. I've taken it from the Pulse database. The chickpeas, the entire thing, sequencing has been taken it from Pulse database. And the rest of the 5 species that I spoke in phylogenetic has been extracted respectively from their databases. Okay, thank you. Thank you. Thank you very much.
The next speak.
Thank you for your presentation, ma'am. For our next presenter, we'd like to call Y Tanvi for their presentation. Yeah. Please join me in welcoming them to the stage.
Tanvi, you have to complete your presentation before 8 minutes, okay? Please emphasize on results.
A very warm good morning to everyone. I am studying CSE Bioscience in the Savita University pursuing the second year. So here I am to present about in silico alliances of Bacopa monnieri and drug development against the bipolar disorder. So first of all, Bacopa monnieri is a herbal plant. So it has many herbs which are useful for the medicine. So mainly it focuses on the pharmacological area. So and by using Bacopa monnieri, I want to focus on the bipolar disorder. So bipolar disorder is a neurodegenerative disorder. So mainly the researchers, most of the teenagers are suffering from this. So I want to focus on this bipolar disorder. So and then so I want to develop, I want to work on the drug. So from the Bacopa monnieri, targeting on the SGP1A1. So this SGP1A1 increase the activity. So it increase the activity and inhibit the phospholipase A2 activity. So the main reason of bipolar disorder is phospholipase A2 activity. So I want to inhibit that. So I have taken the objectives to explore about SGP1A1 and then the drugs I have to work on the Bacopa monnieri. So this is my work plan and I'll explain it to the methodology. So first I have collecting the samples. So and then analysis and which taken from the Bacopa. So I have focused on the ligand and receptor preparation. So after that which I have collected from the Bacopa and then the receptor. So I have done the molecular docking. So from there I got the results and the discussion. So through using eye tracer, I have developed a 3D model for the protein structure prediction. So by using this IT tracer, I have got the 5 models. So by using C-score and estimate TM score and estimate RMSD, so I have chosen a very near 3D model to represent that. So and I have taken the molecular docking. So top 9 places for the binding. So and then this is the molecular function. The overall function I have represented in this molecular chart. So which the main activity is to inhibit the phospholipase A2 activity. So and I have described the human body, so where this will be affected. So the black represents a little minor and the next stage represents the light blue and then the violet, and I have represented to the brain also. So these are the compounds from Bacopa monnieri retrieved from PubChem. I used PubChem to retrieve this. So these are from Bokapa Maneri. So, and then I have did the molecular docking. This was a random docking I have done. So based on the docking, I have whichever the values I have received, I made a flowchart to that. So, and these are the results. So the structures binding in this protein. So I have chosen that. So these three values I felt a little bit closer, so I made it. So, and then we have before in the flowchart, we have seen many compounds of Bacopo Maneri. So from that, so only some compounds like follows the ADME. So we know that if we want to decide a drug, whether it is useful to the human or not, we follow these parameters, ADME, absorption, digestion, metabolism, excretion. So based on this, if a drug follows these parameters, indicates that drug is useful to the human and that is not harmful. So these are the compounds following those properties. So, and then my final conclusion is the overall I have developed worked on the two drugs. So 1234 tetrahydroquinone and trans-2-phenyl, and as mentioned isoquinol. So these are the compounds extracted from the Bacopa Moneri to work on the protein, so to suppress the activity of phospholipids for the bipolar disorder. So so I have studied the short-term efforts of the Bacopa Moneri. In case of the further, there will be the long-term efforts will be the studied. Thank you. And these are my references I have chosen, and that's it.
Questions from audience? Just I have one question.
Yes, sir.
So have you included any positive control in your study?
Like
the drug which is available in the market for this
so
particular
actually, sir, there was any research work has not been done by using Bacopa Moneri on the bipolar disorder. So that's why I concentrated on this. There was no officially included in it in market.
There is no,
No,
drug in the market to treat this disorder?
No, sir.
Are you sure?
Yes, sir, sure.
Then how you
so actually,
design this?
Bacopa is a herbal plant. Use it to treat this neurodegenerative diseases. So it worked on the Parkinson disease, on Alzheimer's. So as that such way, I tried to work it on the bipolar disorder. So I just made it like normal explanation on that. So which was through docking the results. So we included that this drug might inhibit the activity for the bipolar disorder.
Okay, thank you.
Thank you, Tanvi, for that intriguing presentation. Next, we'd like to call upon Shanmugarajan, who will be presenting on interaction of okra proteins with viral proteins encoded by okra inhibition. Please put your hands together, please.
Good morning, everyone. I am Shanmugarajan. I am going to present on the topic of interaction of okra proteins with viral proteins encoded by okra initiation leaf curl virus and okra yellow vein mosaic virus. It's an in silico approach, and my study is completely an in silico approach. First of all, megamovirus, which belongs to the Geminiviridae family, are significant plant pathogens affecting a wide variety of crops worldwide, including tomato, papaya, cucumber, etc. And my study is about okra, which is nothing but a lady's finger, and its scientific name is Albelmosus esculentus. And these viruses are mainly transmitted by whiteflies and cause disease like okra initiation leaf curl virus and okra yellow vein mosaic virus, leading to agricultural crop losses. Okra is an essential crop variety which is known for its nutritional value, including dietary fibers, vitamins, minerals, and it's widely consumed in tropical and subtropical regions. Understanding the molecular basis of these viral interactions is crucial for developing sustainable disease management strategies. And my studies on my studies about the in silico approach, which is interaction between the viral proteins and the plant host proteins, which manipulate the host cellular machinery and establish infections, identifying protein-protein interaction, which could lead to the developing okra varieties and novel strategy drugs. And some of my literature reviews is plant protein and viral proteins interaction studies are crucial for understanding the molecular dynamics between plants and viruses. And in cucumber mosaic virus, viral proteins like 2B interface host RNA, which is known for its RNA silencing, and which could lead to the degrade of viral RNA. And in tomato leaf curl, yellow leaf curl virus, Begoma virus, we have been managed through transgenic plants, exposing viral code protein and triggers resistance. And ELISA technique were employed to develop and detect viral proteins in infected plants. And my study is a novel study which includes okra albumosus esculentus, which has been those genes have not been annotated yet. And my this study suggests that in plant protein and viral protein interactions, understanding it plays a crucial role in understanding the viral disease management strategies and developing a resistance to transgenic crops. And my hypothesis is plant viral protein interaction studies are designed to explore and test how specific plant proteins and viral proteins interact during the viral infection cycle. These studies helps in better understanding and how the virus exploits a host machinery and plant defenses activated or suppressed. And my objective is about the selection and modeling of okra host proteins and viral proteins encoding okra Anatian leaf curl virus and okra yellow vein mosaic virus. And we retrieve the protein models from NCBA database, and the models are evaluated by Ramchandran plot analysis and followed by the plant viral protein docking and interaction analysis were done. And here is the detailed work plan and methodology. Plant protein, first we took about the plant protein and viral proteins from NCBA genome database, and 3D modeling were done by using Robeta server from Baker's lab. And the alpha fold structures of selected proteins were evaluated by Ramchandran plot analysis by using ProCheck server. Potential protein binding sites were identified by using a deep spread web server, which is developed by IIT Madras. And plant protein and viral protein docking was done by Haddock server. And the protein-protein docked complex were analyzed by using PDB-sum. And coming to the result part, after analysis of the literature, we took about the actin, the plant proteins, the actin, cytochrome, B6F, and NADPH, guanine oxidoreductase subunit, MYB, and photosystem 2CP43 reaction center protein. Based on the literature, we took these proteins and the viral proteins from okra Anatian leaf curl virus and okra yellow vein mosaic virus, which shows that AV1, AV2, AC1, AC2, AC3, AC4, as follows by AV and AV1, AC1, AC2, AC3, and AC4 for okra yellow vein mosaic virus. And the alpha fold structures are modeled by in the PDB format and submitted for Ramachandran plot analysis, which shows that less than 5% residues in the disallowed region were taken for further analysis, and more than the results, which shows more than 5% analysis were excluded. And deep spread server awarded a score for 1, that is for active protein binding regions for 1, and 0 for non-binding sites. And objective 3 is nothing but the deep spread protein, that is the active binding sites proteins were taken for the Haddock submission, and which denotes a stable interaction, which it actually shows a positive and negative scores. Negative scores shows that good and stable interactions, and positive scores are which shows the less and negative interactions. Here are some results or the ad hoc score. We only represented the ad hoc score because of the slide limitations, and other van der Waals energy forces and other parameters are not mentioned. And we can see that actin, cytochrome, and NADPH shows high negative interactions between the okra begomaviral protein and okra plant protein and begomaviral viral proteins. And here are some illustration images which actin and plant protein and okra plant protein and viral protein interaction images. And coming to the discussion part, good and stable interaction are found between actin, cytochrome, and NADPH, as I already mentioned. And weak interactions are seen on MIB protein and photosystem reaction center protein. Actin proteins interact with begomavirus, play a significant role in intracellular movement and systemic spread, where the begomavirus utilize host actin cytoskeleton and facilitate a viral movement. And cytochrome B6 complex is a crucial part which plays a major role in electron transport chain in chloroplast of plants involved in photosynthesis. In this case, tomato yellow leaf curl virus changes in expression, chloroplast related genes are observed, and the best and stable proteins from of cytochrome B6F are AV2 and AV. And NADPH oxidoreductase proteins that is say code protein of tomato leaf curl New Delhi virus, a begomavirus, which the code protein gene were actually coded with the tomato plants, so that the viral components cannot occupy the coding region. And the scope of the study is that protein-protein interaction. This research seeks to identify crucial host proteins targeted by begomaviruses, shedding light on how these interactions impact normal cellular process and escape plant defense mechanisms. And additionally, we provide insights and gain development and antiviral compounds and specifically disturb critical viral host interactions. And by this study, we develop transgenic plants and the drugs targeting to the these viral infections. And here are some references. Thank you.
Any questions from audience? Okay, I have one question.
Yes.
So, out of many protein you listed,
Yes,
plant
sir.
protein you listed, if you want to modulate for virus resistance, which one will be a best candidate gene?
From my literature, actin is the best protein against viral infections, sir.
Actin is a housekeeping gene.
Yes, sir.
So if you want to modulate housekeeping gene, do you think you have any other impact?
I think the actin is the best protein for the against the viral infections, sir.
Against where?
Out of 6 genes, which one you feel is viral genes? Begomaviral genes. You have taken
Yes,
6 viral genes.
Yes, ma'am.
To
That
see the
AC1,
interactions.
AC2, AC3.
Yeah. Which one is
Actually, AC1, AV1 and AV2 is a AV1 is a code protein gene, and AV2 is a pre-code protein gene, ma'am. And AC1, AC2 are replication enhancer proteins and transcriptor activated proteins. And AC4 is a movement protein, ma'am, which helps
Which one is highly interacting with the MYC?
that is AV1, ma'am.
AV1.
Yes.
Okay. And one small correction. Don't use AV1, AV2 because this is a monopartite begomavirus.
I'm sorry, ma'am.
You should not use AV1, AV2 for the genes because this is a monopartite begomavirus. Okay?
Okay.
So you can use V1, V2.
Okay. Thank you.
Thank you, Shanmugarajan, for your articulate presentation. Next, we shall move to the next presenter, Sendil Kumar Sricharan, who will be presenting on foliar application of bioregulators improved psychological traits and wheat yield under terminal heat stress.
Respected chairpersons of the session, other professors and other presenters and audience, I am Sricharan S. from Division of Plant Physiology, Indian Agriculture Research Institute, New Delhi. I am going to present on the title, Foliar Applications of Bioregulators Improve the Physiological Traits and Wheat Yield Under Terminal Heat Stress. So coming to the introduction, as we all know, wheat is an important cereal crop next to the rice, and it is also referred as a king of cereals. And this accounts for dietary intake of 36% of the world's population. So with this, its demand keeps on increasing year by year. However, the yield of the wheat reduces and it also lags behind the required 2.4%. This is attributed mainly because of so much of abiotic factors, especially the high temperature and drought. Within the high temperature stress and the drought, high temperature stress accounts major drawback for reducing the yield of the wheat crop. So speaking about the Indian scenarios, wheat is mainly cultivated as a rabi season crop in northern part of the India. So it is started cultivated from the month of November. The sowing starts by the month of November and harvest will be by the end of February. But because of the cultivation of rice, there is a delayed sowing of the wheat crop, which is generally practiced by majority of the farmers in the northern part of India. So once the sowing is delayed instead of November to the month of January, the crops reach the stage of anthesis by the month of March, and the grain filling continues by the month of March, and harvest starts by the April. So meanwhile, when the crop reaches the anthesis, the temperature in the northern part of India gradually rises, and in my case, the temperature has reached up to 40.2 degrees Celsius. So this is termed as the terminal heat stress in wheat, and this accounts for the major yield loss in wheat. So heat stress, the main symptom of heat stress, it induces the premature leaf senescence and forced maturity, which drastically reduces the yield. So naturally, plants have an adaptive mechanism called the stay-green trait. Stay-green trait is generally referred as the ability of the plant to maintain functionally higher chlorophyll content and photosynthetic activity after anthesis, so that it converts the photosynthate into the yield. So, and many literatures have reported that the use of bioregulatory molecules, especially under high temperature stress and other stress, abiotic stress, has found to increase the water use efficiency, proline accumulation, chlorophyll content, relative water content, membrane stability index, antioxidant activities, heat shock factors, yield traits, and also finally the yield. It is also reported in many crops like starting from rice, pulses, and also in roses. So we hypothesize that heat stress induces leaf senescence and forced maturity, which results in poor yield. So biosignaling molecules has been reported to improve the photosynthetic activity and also to mitigate the abiotic stress. So to the best of our knowledge, this is the first study which has been reported to use of gamma aminobutyric acid along with cytokinins, especially benzyl adenine and thiodiazouran, to increase the physiological traits and high heat tolerance and yield in wheat. So we objectivized to explore and dissect the cytokinins BA and TDZ along with gamma aminobutyric acid GABA modulating the stay green trait for heat tolerance and yield in wheat. So this is the experimental plant. We have selected two genotypes. One is Chiria 3 and HD2329. Chiria 3 is already a known stay green variety and HD2329 is a non-stay green type. These two genotypes has been screened from 32 germplasms and we have identified both as stay green and non-stay green respectively. The experimenter was designed in factorial completely randomized designs with two sets of treatment. So the main treatment was normal zone, which was done by the date of November 11th, and late zone was done to expose the crops to the high temperature stress at the time of anthesis. So late zone was done by the month of January, 4th January. So the sub-treatments were application of this bioregulatory molecules at anthesis stage. So water as the control, benzyl adenine, TDZ, gamma aminobutyric acid, and the combination of BA with GABA and the combination of TDZ with GABA. So coming to the results, in our case, under normal zone condition, that maximum temperature ranged from 24.5 degrees Celsius to 34.5 degrees Celsius. However, in our late zone conditions, the temperature reached up to 40.8 degrees Celsius. So this shows the leaf senescence percentage. So we have taken observations on weekly intervals starting from 0 days after anthesis till 28 days after anthesis till the crop matures. So coming to leaf senescence percentage, under normal zone conditions, all the treatments showed maximum percentage up to 80% of leaf senescence by the end of 28 days. However, under late zone conditions, maximum treatments and the control reached 100% of leaf senescence by end of 28 days. Especially with respect to control, our application of bioregulatory molecules, especially this BA with GABA, has found to reduce the leaf senescence across all the days. And coming to spade value, spade is an indirect measurement or non-destructive measurement of chlorophyll content in the plant. So spade value is found to generally decrease starting from anthesis till the crop maturity. And in both varieties, under normal and late zone conditions, our application of BA plus GABA has found to maintain higher spade values. And speaking about canopy temperature depression, in both the cases, in both the varieties, CTD has found to be slightly increased and then started decreasing throughout the days after anthesis. And BA plus GABA treatment also maintained and also improved the canopy temperature depression, especially at day 7 after anthesis, and it also maintained throughout the days. This is a cell membrane injury index, cell membrane stability index. Generally, during the stress condition, the membrane stability, the plant tends to lose the membrane stability, and this biomolecular treatment application has found to maintain or improve the membrane stability index in both the varieties under normal and late zone conditions. So we have also measured the photosynthesis and its associated rates like transpiration rate. Photosynthesis rate and transpiration rate as both has found to follow a decreasing trend throughout and starting from 0 days till crop maturity. But this application of BA with GABA and also TDZ plus GABA has found to improve and also maintain the photosynthetic traits, photosynthesis rate as plus the respiration rate, transpiration rate when compared to the controls. So we have also quantified the chlorophlase enzyme activity to measure the stay green trait. Chlorophlase A and also chlorophlase B has found to increase with increase in days after anthesis, and the control showed the highest activity of chlorophlase enzyme. However, BA plus GABA treatment and TDZ plus GABA treatment has found to reduce the activity of chlorophlase so that the chlorophyll degradation is reduced. So we have also conducted an RT-PCR expression that our application of BA plus GABA, especially on 14 days after anthesis, has found to increase the expression of Rubisco larger subunit under normal and late zone condition in both the varieties. Coming to yield traits, under normal zone condition as well as under late zone conditions, control is found to have the lowest yield, and when comparing between the sowing seasons, late zone season has found to drastically reduce the yield because of the high temperature, and application of this BA plus GABA treatment has found to improve slightly a better yield as compared to the control plant under both the varieties, under both the cases. So to conclude, this bioregulatory molecule, especially this BA plus GABA treatment, has found to increase the heat tolerance in wheat, and also it has reduced the heat susceptibility index and also yield stability index in wheat. So we conclude that this application of BA plus GABA with and this TDZ plus GABA helps wheat under heat stress by delaying leaf senescence and improving physiological traits. So this can lead to the better canopy cooling and stable yields. So it shows a promise for developing heat tolerant varieties, and it is also a direct use for the farmers in the societies. So this is the plant Chiria 3 and HD2329 under high temperature stress conditions. So we can see a clear-cut difference because of the application of BA plus GABA under both the cases with respect to plant height, the tiller number, density, the greenness, and also the ear length and the weight. So I would like to acknowledge my chairman, Dr. Pramod Kumar, our abiotic stress physiology lab, all my advisory committee members, the Division of Plant Physiology for providing the facilities, Indian Agriculture Research Institute for the financial support, and Department of Genetic Engineering for organizing this conference. Thank you.
Any questions from audience?
Yes. Yeah, it is. Yeah, yes, sir. It is already well known, reported, but we focused only to improve the stay green trait by help of the cytokinins. Normally, under high temperature condition, the activity of cytokinin oxidase is found to be more, and it reduces the level of cytokinins, which is synthesized within the plant. So we found that exogenous application of the cytokinin molecule can improve the stay green rate under high temperature. And also with respect to GABA, it is already increasing under abiotic stress, but we feel that that amount is not enough for the plant to cope up this stress. So we have just focused on bioregulators, especially this two cytokinins and GABA. Thank you.
Thank you for your detailed presentation, Sandil Kumar. And for the next presentation, we'd like to call upon Atreyi Mahinder on the topic Identification of Potential Inhibitors of Tumor Suppressor Gene from Phytochemical Constituents Found in Tomato Solanum type Icopyrsum via Biocomputational Analysis. Please welcome them onto the stage.
Good morning, everyone. I am Atrey Majumdar from Techno India University, Kolkata, West Bengal, and my guide name is Malavika Bhattacharya. And we presented our topic is identification of potential inhibitor of tumor suppressor gene, that's P10 gene from phytochemical constitution found in tomato Solanum lycopersicum via biocomputational analysis. What is happening? First introduction, the tumor suppressor gene, P10 gene. P10 gene is first identified in 1997. It's phosphate and tensine homolog gene. That this tumor suppressor gene is chromosome 10, and its inactivation makes the different types of cancer like endometrial cancer, lung cancer, breast cancer, prostate carcinoma, ovarian cancer, etc. And it's regulate the PI3K AKT pathway by phosphorylation of PIP3 at the cellular membrane, and loss of operating P10 protein results the tumorigenic PIP3. So lots of studies I have concluded that the 30 to 80% P10 gene has been actual reasons of the endometrial cancer. And the endometrial cancer is it starts in the endometrium where where they start to proliferate out of control, and in develop the tumors in the uterus. Endometrial gland developing cells are where the cancer has been started. Endometrial carcinoma is thought to be caused by several genetic changes like including signaling system disruption, proto-oncogene activation, tumor suppressor gene like P10 inactivation, etc. and also unopposed estrogen and development of endometrial hyperplasia and hormone receptor activity that change the genetic of the molecular processes of oncogenesis. This is a review of literature I have taken for my work. And the hypothesis of my study is to perform the potential inhibitor of tumor suppressor gene related to the phytochemical constitution found in silico analysis, bioactive compounds in the pulp, peel, seed of the tomato extracts, and the epidemiological evidence has been ingestion of the tomatoes. And my objective is to evaluate the evidence produced and investigate the potential advantages of the bioactive compound present in the peel pulp cells in the tomatoes. So molecular assessment of the method of the biocomputational techniques, factors controlled by through the data analysis of docking, and the possibility of residual uncontrolled confounding. My work plan, the gene expression profiling by the in silico work assess, identify interaction between P10 gene and bioactive compounds of Solanum lycopersicum and its effect in P10 gene by the different tools of bioinformatics method, preparation of target protein, analysis stereochemical properties of the target protein, retrieval the ligand found in peel, pulp and seed of the tomato, molecular docking analysis, and analysis of the suitability of ligands as potential drug targets. And of all this I have compared with the lenimative capsules. This is called lenimative mesylate. That this drug is used in the endometrial cancer, and the potentially beneficial properties of phytochemical constitution retrieve the tomatoes, and then compared to the same benefits of this prescription drug, and the possibility of developing any drug, the implication utilizing phytocompounds found in the endometrial cancer inhibiting the hallmark of P10 tumor suppressor gene has been discussed. So my methodology is the P10 tumor suppressor gene has been obtained from the RCBC PTV web servers and in the 3D crystal format. And it is noted that the crystal resolution 2.10 Armstrong single chain of the 324 amino acids has been US CF Chimera software has been docking studies following the standard protein. And analysis of these radiochemical properties of this I have done the preparation of target protein, and now analysis of the stereochemical properties of target protein. The three-dimensional stage of target protein of the P10 tumor gene, the PDB ID, the ID 5R has been I have studied, thoroughly understand its stereochemically, and the EMBL EBI PDB suit cited or ProCheck web server, I have taken this P10 gene. The retrieval of the control drug ligands found in peel pulp and seeds in the tomato. I have of the peel pulp and tomato seeds of tomato. I have taken the PubChem database and potential inhibitors of the P10 suppressor genes. I got the in the tomato peel is 55 phytocompounds, tomato pulp is 37 phytocompounds, tomato seeds is 225 phytocompounds, and my control drug is lenimative mesylate. And I have taken its 3D SDF file format, and this phytocompounds has been prepared by the ligand minimization energy and P10 inhibited or the lenimative mesylate, that methane sulfonin sol obtained by the reaction of lenimative with the molar equivalent methanosulfonic acid. So so in the comparisons, I have done the molecular docking and the Pyrex software, Python prescription software I have uses, and I have got the chart, and also the admin analysis has been done, where I have done the rules of 5 drugs development, the Lipinski rule of 5 drugs development, where he told that no more 5 hydrogen bonds donors, no more 10 hydrogen bonds acceptor, molecular weight less than 500 grams per mole, molecular reactivity between 40 to 130, and value of the log P and log C is less than 5. So I have got this binding energy with the 1D5R P10 gene, where combatin is minus 9.8, flavonone is minus 8.8, where the lenivative mesylate has minus 8.4. Also, cryptochrome is minus 10.2. We know that minus which be more is it good binding energy is can be a good drug can be. So, but in the Lipinski rule of 5 rules, the combatin and cryptochrome failed. Combatin failed 3 times, where the molecular weight is higher than 500, H bond donor is higher than 5, log P is very less, molecular refractivity is less than more than 140, and also H bond acceptor also more than 12. So now the boiled egg analysis I have done for the drug likeliness, where the compounds have been in the white portion is less drug likeliness, where is human intestinal absorption has been happened, and the yellow portion where the blood-brain barrier, where the drug likeness is more. And I have done a Swiss ADME analysis, and it's done also. It's a help of Whiteman topological polarized subdivision area. So in the result and a discussion where the admin analysis and boiled egg analysis of the the pharmacokinetics had been and the toxicity testings, their combatin has a accuracy is 100%, where lenivative mesylate has 67.38%, but all the log is the polarity, glycoprotein, human intestinal absorption, blood-brain barrier, and carcinogenity, combatin and cryptochrome has been rejected, but flavonone, naringenin, solanocol has been accepted. So, so we can furtherly can be herbal drugs, and we can change from about the lenivative mesylate. So scope of the study, further research has been distinguished the phytochemical constitution of bioactive, improve the stability of the before the drug candidates, and and my P10 gene has been activation in the suppressor of the P10 tumor and metastasis. In the conclusion, its study revealed that the peel pulp seeds of tomato has been combating cryptochrome is more, but naringenin, flavonone has same. So in this phytocompounds of the other bioactive components of the potential drug candidates against target protein P10 tumor, we can use this this naringenin and flavonone, etc. So this is my reference. Thank you. Thank you.
Questions from audience?
Only limited one from pulp, peel, and seed.
Actually, I have studies the paper and peel pulps studies. There is so many papers is there. The peel pulp and seeds has a very good herbal production. Actually, I have studies the in the corona time. I have studies the tomato is a very good for the. And that time me and with my guides are doing research for the tomato if we select, and then in that time I got this study materials actually, and then in silico analysis I've done.
Yeah, fine. Thank you.
Thank you so much.
One more question.
Hello. Hello. Thank you for the presentation. It was very nice.
Thank you so
and
much.
I just wanted to ask, what is the need of blood-brain barrier? Like, why did you check for a blood-brain barrier here?
Because every drugs not been absorption with the intestinal, also it has to be the blood-brain barrier. So we just checking that, and not so many. I have data as is there. So just we did the boiled egg analysis and got this.
So you're saying like for any drug that we have to test, it has to pass the blood-brain barrier for anything that we are taking. It has to pass the blood-brain barrier. That's what you're saying.
Yes.
Okay.
Thank you, Atreyi, for your presentation. And for the last presentation, we have Reena Chaitri, who will be presenting on the topic From Tradition to Therapy, Anti-Inflammatory and Anti-Alzheimer's Potential of Selected Ethnomedicinal Plants from Sikkim, Himalayas. Please join me in welcoming them to the stage.
It's PDF. Good afternoon, everyone. Respected judges, all the dignitaries on the dais, off the dais, all my fellow mates, and my audience. I'm so glad to be here. This down to the last section of India for the first time. Thank you for giving me this opportunity. So as today I'm here not to express the critical aspect of medicine, but also the traditional knowledge that my grandfather, forefather have been using for treating the life-threatening disease for generation. Here comes to the focus my today's presentation, that is from tradition to therapy, anti-inflammatory and anti-Alzheimer's potential of selected ethnomedicinal plants from Sikkim, Himalaya. Okay. This ethnomedicinal plants plays a very important role in the people of Sikkim because of the geography, the topography, the area. It is so remote that they do not find the facility for the allopathic medicine. So for them, this ethnomedicine is very important whenever there is a need. So for today, I am talking about those people, traditional people who has been using those medicinal plants from the Himalaya to treat the cognitive diseases, basically to the focus of Alzheimer's disease. So before understanding Alzheimer's disease, I would like to explain something about dementia, that it is an umbrella term that say it is a term for the loss of memory, thinking, and abilities severe enough to hamper your daily life. So there are many types of dementia. There are Alzheimer's disease, Lewy body dementia, frontotemporal dementia, vascular disease, and any other dementia, mixed dementia as well. But above all, the most prevalent is Alzheimer's disease. So this is the most prevalent form of dementia. The 60% of the dementia is due to Alzheimer's disease. So it also, why Alzheimer's disease? Because as people do not understand in the hilly area what Alzheimer's disease is, so they treat the symptoms of this disease that is characterized by the progressive impairment in memory, learning, behavior, and the dysfunction in the daily life. So drug development in Alzheimer's disease is faring higher failure rate. Till date, there was no such existing report for the treatment of Alzheimer's disease. So I was there to document and scientifically validate the traditional medicine that they are using for generation really have the potential to treat or not. So these are the literature review shows that there are so many therapeutic strategies applied by the researcher to treat the Alzheimer's disease. For now, I am taking the antioxidant, anti-inflammatory, amyloid beta, and then cholinesterase. For now, I am in the middle of my studies, so I completed my phytochemical, antioxidant, anti-inflammatory, and cholinesterase. So I hypothesize that in recent years, herbal medicine have played an important role and get great attention and acceptance worldwide because of low efficiency, low cost, promising efficiency, and lesser side effects. So medicinal and aromatic plant plays a very crucial role in the people of Sikkim. So this ethnic population of Sikkim may hold the tremendous amount of knowledge on ethnomedicinal plants. So does the plants they are using for the treatment really have the potential to treat? So I hypothesize that medicinal and aromatic plants of the Sikkim Himalayas may have the potential to treat the Alzheimer's disease and related dementia. So my objective is to identify and document the ethnomedicinal plants and then to scientifically validate. So these are the work plan and methodology. I went all the way to the remotest area of Sikkim to meet the healers. They are practicing to the real, the whole people. They are without tasting it. They were using. So it was very marvelous for me. So I went all the way to them, and then I collected the data, and then I identified the plant, and then after that I did the phytochemical in vitro analysis such as phytochemical, antioxidant, anti-inflammatory, in vitro cholinesterase and activity, and later on it was the other section, virtual screening is there, and then the in vivo is also there because it is funded by the ICMR, so there are a series of experiments to be done. So till now I have done this. So my results are the phytochemical analysis I have done, but not put the graphs here. These are the graph prepared by the using origin, the antioxidant by using the ABTS analysis, and then DPPH, and then I've got the highest antioxidant potential of this allium oligochaete methanolic root extract, and all this result shows the best on the methanolic extract, and then ABTS, IC50 to the also, and then to the standard molecule, that is ascorbic acid, also the inflammatory activities using hydrogenase, and then 15-LOX activities, and the highest was also the from the Myconopsis paniculata, it is 34.66. Likewise, there is the sorted 8 plants is, I have done the acetylcholinesterase activity, and later on the 12 of the sorted plants from the phytochemical, we have done the GCMS from here itself, LCMS also, and then few molecular docking studies I have shown over here. But there are so many, so these are the whole work. So results that are out of the interviews, I have identified and documented 56 plants, and 30 have showed the high amount of total phytochemical contents, and then 13 are again, 13 I did for the anti-inflammatory, and then 8 with all the 13 for antioxidant activity, ABTS and DPPH, 8 are sorted out of that with the best below 100 microgram per ml IC50, and then so for 8 I have done the anti-inflammatory and acetylcholinesterase activity. So the scope is that as this work is for the first time, I've done from my state identification of promising medicinal plants as they are dependent on it, and then they are using on the people around them, and they are trusting it. So, and then quantification of bioactive compounds, and then evaluation of in vitro anti-inflammatory activities, and also towards the Alzheimer's disease, because they were promising the patient that it will treat. So I tested in the cholinesterase activity, and then docking study, so whether they have the property or not. So these are the references I've done pointed out. Thank you so much.
Any questions from audience?
Have you used any other solvents for extraction apart from methanol?
Sir, methanol, hexane, and water I have used. Yes, sir. So methanol shows the highest activity, sir.
What is the reason?
Because it is a polar molecule, I think. So other than that, it has been always been the methanol, sir. I have found that, sir. I found one interesting that water does have, with the water extract, I have got the value, but not with the other, not in other analysis, only in the phytochemical. So that's why, sir.
So 56 plants you
Yes.
have collected. So you have identified all the plants.
Yes.
The,
Yes. It was all Himalayan plants, ma'am. It
Hmm.
was above the 300 meter above. So,
Okay.
yes, ma'am.
Is it, how easy it is to collect the plants?
Ma'am, prior permission from the forest department was required. As I am from that place itself, it was easier for me. So I went all the border area. It is nearby China, and then we collected all the samples from there. And then it was not too much. I just had to collect a few so that I could analyze, ma'am.
Okay, thank you.
Thank you. Thank you so much.
Thank you for all your participation and enthusiasm. Today we conclude the oral presentation session. So if any of you are participating in the poster presentation in the domain of microbes and plants, it's parallelly happening in biotech lobby. So students are kindly asked to move to the biotech lobby if any participants are here. I just I am delighted to welcome you all back to the continuation of lightning competition. Yesterday we witnessed some incredible presentation where participants showcased their research with clarity and conciseness. Today we have yet another exciting session lined up featuring more brilliant minds ready to share their work in just 3 minutes. Before we begin, let me remind you the rules. Each participant will have exactly 3 minutes to present their research in 3 slides and without any animations and video. So I would like to welcome the judges for this session, Dr. Aruljyoti Nagarajan and Dr. Ganeshan. Thank you for being here. So let's begin. Our first presenter of the today is Saranya E, SRM Institute of Science and Technology.
Good afternoon, everyone. This is Saranya E, research scholar in SRM-IST. So my research entitled Label-Free DNA Enzyme-Based Colorimetric APTA Sensor for the Detection of Leptospira interrogens. So aptamers are single-stranded DNA oligonucleotide that will have a 3D conformation to bind to its target. So we have isolated a specific aptamer sequence for a pathogenic Leptospira interrogens species. And the aptamer, usually it will be coated onto a nanomaterial for it to detect the pathogen in the system. Here, instead of using nanomaterial for the signal transduction, I'm using a DNA enzyme sequence, which is an 80 nucleotide sequence that can mimic as a peroxidase enzyme. So aptamer plus DNA enzyme sequence will give you the aptazyme, which is the sensing moiety in the system. So moving on to the methodology, the aptazyme sequence, it has to be coated onto the carbon nanotube in order for the aptazyme sequence to remain in off stage. That is, it should not produce signal in the absence of leptospira interrogens. So after it is coated onto the carbon nanotubes, then it will remain in off stage, and then when the leptospira interrogens, that is the target pathogen, enters the detection system, the aptamer domain in the aptazyme will come out and bind to the leptospira interrogens. Then the DNA zyme sequence, which mimics as the peroxidase, requires a catalyst, which is hemin. Once the hemin is in the G quadruplex structure of the DNA zyme sequence, it will act as a peroxidase enzyme, and it will convert the peroxidase substrate, that is H2O2, with a chromogen, that is ABTS. It will turn the colorless ABTS into a colored compound, which is in green color. So here I have shown some results of the sensor, sensitivity and specificity, and validation with other reservoirs of leptospira. So sensitivity, I gradually increase the concentration of the leptospira interrogens from 0 to 1 into 10 power 5 CFU per ml. So with the increase in concentration of the leptospira, the color intensity also increases. Moving on to the specificity of the system, the aptazyme did not produce any color in the presence of other species of other bacteria, that is E. coli, Pseudomonas, Proteus mirabilis, all that. And the ninth well, the ninth tube, that is the positive control for the system, that is, it has only leptospira interrogens. Only in that tube we have color. The validation is, there are around 240 reservoirs present in leptospira interrogens, and there are other pathogenic strains as well. Leptospira Kirchneri is one of the strain that is rare in human, but our system gives color for that also, and I have presented result in C. So the comparison of this system with other methods, that is RT-PCR and PCR. So PCR, we have diluted the culture solutions in 10 power 7 to 10 power 3, and I've presented the result. Comparatively, our system also works till 10 power 3 CFU per ml.
Thank you, Saranya. That was a wonderful talk. And up next, we have Kumaran K from SRM Institute of Science and Technology.
Hi, good afternoon, everyone. My topic is like identification of monogenic and polygenic cause of familial hypercholesterol in Tamil populations. Like you can see, like Saurav Ganguly is a famous cricketer and Bollywood actors. They are getting the myocardial infarction in age less than 40. So the one of the main reasons is hypercholesterolemia. So hypercholesterolemia is one like which is a major consequence of premature CVDs. So autosomal dominant disease. Like it is caused by the monogenic genes of LDL upper BPCSK9. Along with it, there are some 64 gene loci available. See, like in the 5 years ago, there are like 1 in 500 FH prevalences, but that has changed to 1 in 150 in 2024. So, like we need to address this gap. So the gaps, there are many gaps, and the cascade screening also very important for this. So my aim is to identify the variants associated with the familial hypercholesterol in the Indian population. So using first, I regulate the patients according to LDL cholesterol greater than 155 based on Dutch lipid network criteria, and I will collect the patient blood sample, genomic DNA isolation, followed by analysis of mutation using HM analysis and clinical exome sequencing. So first, I started with the clinical exams like HM analysis. So in this, I have got only like polymorphisms in the LDL gene. So we shifted to clinical exome sequencing to identify the novel variants. So I will be targeting for a immunosequencing lipid metabolism genes. So analyzing using immuno-based phase. So totally 104 patients have been collected. So like definite PFH, I got 6 patients. That patients I got the clinical exome sequencing. So this is the Venn diagram, the common genes present in the all the patients, and followed by like when you found it, there are genes like next genes like PP2R2B, like PMM1, ATX, N2 are responsible other than the lipid metabolism genes. So the FH, basically the lipid metabolism genes are also involved in neurological disorders, cardiovascular diseases, cancer biomarkers, immunosuppression diseases, liver and kidney disorders. So the importance is like we need to establish the entry in the national FS database. So what you're finding, like we need to submit so that it will be world better. So what's my conclusion is first understanding myself, like what's the work, then novel genes of the disease, awareness of FH. So my conclusion is research is not about the finishing thesis, but also like we need to contribute to the world. So whatever work you are doing, it should be helpful in finding novel genes for the world safety. So my work will be guided. I thank you, Dr. Kane Aljuri, my guide, my inspiration, and these are my lab mates. Yeah, thank you.
Thank you, Kumaran. Up next, we have Lakshmi from SRM Institute of Science and Technology.
Good afternoon, everyone. This is Lakshmi S from SRM. So I'm going to present my work on nanoparticle encapsulated with fluoroclusional against gastrointestinal cancer cells and cancer stem-like cells. Gastrointestinal cancer ranks the top 10 in cancer incidence rate in Asia. Cancer stem cells are involved in cancer recurrence and chemotherapy resistance. Hence, we aim to target both the compounds with fluoroglucinol, which is a phenolic compound. So this compound is used in treatment of the ulcer and irritable bowel syndrome, but it has a limitation such as poor water solubility. Hence, we encapsulated it inside a HAMSN nanoparticle. So it is a series of process by which we synthesize. After that, we characterize and then we perform the drug release kinetics. In that, we observed that our nanoparticle was able to release the drug only at a very low pH and higher concentration of GSH, which is highly important for the targeted drug delivery. Then we perform the basic toxicity assessment using hemolysis, toxicity in normal cell line, and in vitro zebrafish toxicity assessment, where we found that our nanoparticle is safe and non-toxic. Then we perform the anti-cancer activity. For this, we chose the colon and the gastric cancer cell line because ulcer and irritable bowel syndrome form the basis for these two cell lines. Then we performed the cellular assays. In the cell proliferation assay, we found that our nano carrier HMSN did not show any cytotoxicity, whereas our MSN PGHN nanoparticle was effective than the free fluoroglucinol. Then we performed the cellular uptake assay, where we observed that our nanoparticle was able to undergo HA receptor-mediated endocytosis. Then we performed cell migration, colony formation, and spheroid assay, where we found that the nanoparticle was more efficacious than the free fluoroglucinol as well as the control. After that, we calculated the apoptosis induction using AOEB, DAPI, Caspase 3/7 expression, followed by the MMP evaluation, where we found our nanoparticle was more effective than the free fluoroglucinol. Then we performed the network pharmacology based study in order to find out the molecular mechanism, where we did the ADMET analysis, DFT calculation, shortlisting of the top 10 genes, docking and simulation study, where we found that PG have higher interaction with the GLY1 protein. Then we performed the correlation analysis, and where we found that GLY1 has more correlation with the SMO protein. Then followed by we did the expression analysis, in the gene as well as in the protein expression analysis, we found that our nanoparticle was able to downregulate the GLY1, SMO, and cancer stem cell marker CD44 more effective than the free fluoroglucinol. Hence, we conclude that our final nanoformulation was effective in controlling the cancer cells as well as the cancer stem cell proliferation. Thank you.
Thank you, Lakshmi, for that presentation. Next, we have Karthikeyan from SRM Institute of Science and Technology.
Okay, good afternoon, everyone. Myself, Karthikeyan, working under the guidance of Dr. Ratina Sabhabadi from the Department of Engineering Engineering. And my today's topic was novel quantum dot aptar sensor for the fluorescence turn-off, turn-on aptar sensor for the detection of Vibrio cholerae O139 samples. So, you all know about Vibrio cholerae. It is a waterborne pathogen. It causes cholera. And over the cholera is heard about over the many decades. Cholera is still not a negligible disease. Still, humans are affected about cholera. And the major available methods for detection of cholera is mainly three methods: the serological method, the molecular method, and the culture-based method. And these methods has its own drawbacks. Like the molecular-based methods are highly time-consuming and there's infrastructure and need professional people to do the work. So I simplified my biosensor to detect on-field detection of Vibrio cholerae O139 in remote areas, and it need not requires any express position. And then for that, I choose my biosensor which is made up of aptamer. So it's not moving. So coming up with haptamer, aptamer is nothing but a single stranded oligonucleotide. The unique property of aptamer is it will fold into a 3D structure like jigsaw puzzle pieces. So I incubated my many aptamer sequences with my Vibrio cholerae sequence. Only the sequence that are has the affinity with Vibrio cholerae attached to the Vibrio cholerae. The non-affinite cells are washed away. The only the aptamers with has the affinity to Vibrio cholerae with attached it. Then I PCR amplified my aptamer sequences. Then the PCR amplified sequences are highly affinity towards the Vibrio cholerae. So to know the exact jigsaw fossil for my Vibrio cholerae sequence, the aptamer is highly specific for Vibrio cholerae only. Next, the transducer part. The biosignature consists of two major components. One is the organic ligand and the transducer. The transducer is nothing but a nanoparticle. So I synthesize quantum dots. I synthesize my quantum dots out of Masambi peel through hydrothermal synthesis. So I conjugated my aptamer on the surface of my quantum dots, and I used graphene oxide as a fluorescence quencher. The graphene oxide will quench the fluorescence property of my quantum dot upon the addition of the aptamer sequences. Once the Vibrio cholerae was introduced into my biosensor, the aptamer has the highest affinity towards Vibrio cholerae rather than the graphene oxide. So the intermolecular distance between the graphene oxide and my quantum dot aptamer is about 10 nanometer size. So the graphene oxide is slowly detached from the biosensor. So the quantum dot regain its fluorescence property, which is showed in the research one. With the increasing concentration of Vibrio cholerae, the fluorescence property of my quantum dots it's regaining. The control has no Vibrio cholerae cells in it, and it is increasing concentration of Vibrio cholerae. And for the selective analysis, I choose Vibrio palameticus, Aromonas hydrophila, MRSA, and E. coli cells. The well contains the Vibrio cholerae O139 cells, only recovering the fluorescence property due to the presence of Vibrio cholerae O139. And that's it. Thank you.
Thank you, Karthikeyan. Up next, we have Chandrasudan from SRM Institute of Science and Technology.
Good morning, one and all present here. So I am myself Chandrasudan, pursuing my PhD under the guidance of Dr. R. Satish. So I am here to present about compound A as a potential therapeutic agent for colon cancer treatment. Compound A is a phytochemical. So since it hasn't got the patent yet, I don't want to reveal it here. So my aim is to target the colon cancer cells and the colon cancer stem cells using the compound A. My objectives are to determine the effect of colon cancer proliferation and migration. The second objective is to validate the compound A by in silico method. And my third objective is to investigate the efficacy of compound A in targeting metal 3 stem cells and renewal of colon cancer. So these are my assays which I'm going to show it. Objective 1, cell proliferation assay, migration assay, invasion assay, CAM, colony formation assay, apoptotic cell cycle, and ROS. Objective 2 is in silico, which are RMST, RMFD, ADMET, and hydrogen bond assay. Objective 3 is RT-PCR and Western blot. So I want to tell about the introduction. What is metal 3? Metal 3 is a methyl transferase protein, which helps in colon cancer proliferation naturally. So I want to inhibit the methyl transferase methylation, so I am using this compound to demethylate or inhibit the methyl transferase. So I came to know about IC50 of 45 micromolar is used to as the 50% population of the cells were dead, and I used the same compound to check the cell viability assay and treated in HCT cells, which are colon cancer cells. And also I overexpressed the metal 3. I came to know that 110% has been proliferation was observed in the overexpression. And I did hemolysis to check the RBC lysis, and using the IC50 value, which is 45 micromolar, and there are no RBC lysis. And did migration assay whether E-cadherin methylation has been inhibited or not. So I did migration assay, and also I checked at the treated there was no migration, whereas in control there was migration. And I did colony formation assay to check whether this compound inhibit the colony formation and or not. Where I saw metal 3 overexpressed as more number of colonies, whereas in treated colonies were inhibited. And this is the graph. and I did apoptotic assay using acridine orange and ETBR. So in treated, I was able to see red color and orange color. Red color are dead cells and orange color are pre-apoptotic cells. So I did immunocytochemistry to check whether this compound is going to bind it at methyltransferase or not. So I treated my compound in the colon cancer. So in the control, I saw methyltransferase protein, whereas in treated, I was not able to see methyltransferase protein. And molecular docking, I got minus 8.31 as affinity, and I did RMST, where deviation was below 2, and fluctuation I did RMSF, where some of the amino acid which are in active site and in the alpha foldings, I saw some of the active sites got fluctuated, and hydrogen bonds I have average of 2 number of hydrogen bonds were 2. So, and I got 4-5 hydrogen bonds at 3-4 nanoseconds. Thank you.
Thank you, Chandrasudan, for that presentation. Up next, we have Mohammad Adil from Dr. ALM PG Institute of Basic Medical Science, University of Madras.
Yeah, this. Can you start? Respected chairpersons, professors, and my dear colleagues, good afternoon, one and all present here. I am Mohammad Adil, first year research scholar from Department of Endocrinology, University of Madras. I'm going to present you my study design, that is assessing the neuroprotective effects of vitamin B12 and gabapentin administration in type 2 diabetic rat model. So diabetes is a medical condition characterized by high blood glucose levels due to insufficient insulin production and insulin secretion. It is a worldwide disorder, and it is a cause of a lot of fatalities. My study especially focuses on type 2 diabetic complications affecting various organs. This study focuses on investigating the neuroprotective effects of vitamin B12 and gabapentin in diabetic rats, which may provide important therapeutic effects and therapeutic insights for patients who are suffering from diabetic neuropathy. Vitamin B12 is crucial for the nervous system, which helps in the assisting of maintenance of nerve insulation and production of red blood cells. Conversely, gabapentin is an anticonvulsant which helps in the treatment of epilepsy, seizures, and neuropathic pain. Okay. The aim to determine the neuroprotective effects of vitamin B12 and gabapentin in type 2 diabetic rats. We hypothesize vitamin B12 and gabapentin may have ameliorative and neuroprotective effects in type 2 diabetic rats. Our objective is to study the ameliorative effects of vitamin B12 and gabapentin on nerve function in diabetic rats, to investigate neuroprotective effects of vitamin B12 and gabapentin on key signaling molecules and redox antioxidant system in cerebral cortex of diabetic rats. So our experimental design we'll have 5 groups, each group containing 10 male star rats of 200 grams per body weight. So the first group will be a control group, will be giving a normal standard rat feed for 120 days. The second group will be a diabetic group, will be giving a high fat diet with 30% fructose for 120 days. And the third group will be a diabetic group treated with vitamin B12 50 mg per kg body weight for the last 60 days. And the fourth group will be a diabetic group treated with gabapentin 50 mg per kg, 60 mg per kg body weight for the last 60 days. And the fifth group will be a combination of vitamin B12 and gabapentin in the diabetic group. So once the treatment period is done, we'll euthanize the rats and we'll see several biomarkers and assays. So biomarkers will be doing a behavioral study to confirm the presence of diabetic neuropathy, such as thermal method, mechanical method, chemical methods, and we'll be seeing we'll do molecular work also. So we'll be seeing Western blot, ELISA, RT-PCR, and biochemical analysis. We'll be doing brain-derived neurotropic factor, nerve growth factor, pro-inflammatory cytokines such as TNF-alpha and IL-6 will be seen. We'll do histological examination of sciatic nerve, cerebral cortex, and pancreas. The ad hoc ESA grant awarded to Dr. R. Ilangovan by Endocrine Society of India is greatly acknowledged for this study, and I'll be starting this study very soon. Thank you.
Guys, this lightning talk is to give practice to the PhD scholars to summarize the work in 3 minutes. Okay, they have prepared. If you guys talk in between, they cannot put it out clearly. Okay, please don't talk. Okay, thank you.
Thank you, Mohammad Adil, for that presentation. Up next, we have Reena from SRM University, Sikkim.
Good afternoon, everyone. Again, I got a chance. Where is it? Okay. To say all the things I have forgotten before, I think. Yes. Now, where is my topic? My topic is from tradition to therapy, anti-inflammatory and anti-Alzheimer potential of selected ethnomedicinal plants from Sikkim Himalayas. You cannot see my title though, but I have written. So I'm sorry for that. But here, as because my topic is so inclining towards the traditional medicine, so I want to say that these medicinal plants from the Himalayas are very crucial for our people, and they have been protecting us. And then the people who has been using this plant do not want to shift to the allopathy at all till now also. So I do really wanted to check whether these people who are using those medicinal plants to the patient from our surrounding really have the potential to treat the mental illness or not. It can be any symptoms. It can be from forgetting to getting the freshness, and then it can be in the any form. So there are so many the community based work I have done. So these are from something is missing. So there are so many glitches in my slide. I'm sorry because my Mac problem. So because there are two stages of my work. One is the community based work, and the one is the lab work. Community based work involves the prior pilot study was done, and then we have gone through all the healers, and then collected their view on the using of their medicinal plants, because one healer used the same plant with other plant, polyherbal. So one healer used another, and then because we have to check whether that is efficient or not, so we did the quantitative indices, use the quantitative indices to verify that. After quantitative indices like relative frequency citation, fidelity level, and informant consensus index, we have identified 56 plants is the best they are really using for the symptoms related to Alzheimer's disease specifically. So after that 56 plant, we have identified the 30 plants have best phytochemical properties, and then we sorted after that the plants which have the IC50 below the 100, then we did the antioxidant activities. I've shown you this before as well, and those plants which are having the best antioxidant properties are from more than 300 above mean sea level, 3000. And then those plants, why? Because I have found that those are in the stress condition, and some of them are covered in the snow most of the time. So that may be the reason for them to be effective. And the healers, I would like to say that healers are not at all wrong in their use, but their doses and their use at the different time varies as per the person to person. So my result shows that yes, the traditional healers using the medicinal plants from Himalayas really have the potential to treat the symptoms of Alzheimer's disease for now. I'm still doing my work. Thank you so much.
Thank you, Reena, for that presentation. And up next, we have Vedashri from VIT. Can I know if Vedashri is here? Okay, that brings us to the end of the lightning talk. A huge round of applause to all the presenters for the remarkable ability to condense their research in just 3 minutes. Their efforts truly highlight how the power of clear and effective scientific communication. Thank you once again for all the judges and audience for making it a successful session. And now we will break for lunch and get back and catch up here in the same hall at 1:40 sharp.
அப்புறம் ஏன்னா யோகாங்க